Daniel did a lot of work in helping to pull this conference together, and we very much appreciate it. I'd also like to thank our conference sponsors, Intel in particular, but IBM, Oracle, and Pharma for helping us put this on today. I'd like to also thank our staff, Health IT Now staff, and specifically Catherine Pugh, Molly Nunez, Jen Kilsticker, and in particular Sloan Salver, who did the yeoman's work in putting together today's events. So I was telling a few people beforehand, we really hope this conference is productive. If you came here for a dog and pony show, you're going to be disappointed. We're going to spend about a third of the day today focusing on what are the barriers to using genomics and data-driven medicine in a productive way. And we're going to spend about 2/3 of the day really talking about how we might knock down those barriers. We believe there are operational barriers, technical barriers, there are regulatory barriers, legislative, legal barriers, and all those should be discussed and addressed. So really 2/3 of the day we're focused on that. Just my 2 cents on the issue, I think it's important to note that since the enactment of the HITECH Act, industry and taxpayers have put about $100 billion on the table to finance the adoption and use of health IT in order to meet America's 21st century healthcare needs. I think these investments have the vast potential for revolutionizing healthcare in America, but unfortunately we have yet to see the results of those investments. I think it's a safe statement to make that knocking down those barriers that largely exist in a pre-data era of medicine are absolutely critical to capitalizing on those investments. I think nowhere is the potential opportunity greater than in genomic medicine. I think the mapping and sequencing of genes to discover and create individualized treatments for genetically driven diseases like cancer and diabetes really holds a key to unlocking the future of healthcare outcomes and policy. I think the emergence of genomic medicine in the U.S. Has us on the threshold of a golden age of cancer treatment in particular. And I think cancer, everyone knows, has a friend or relative who's been impacted by cancer. I know I have just this year lost a close relative. That cancer is the era of genes and gene duplication. And so the solutions to cancer are really an individualized look at how to sequence and address those errors in gene duplication. And so the treatments that ultimately will cure cancers, because it's not a singular disease, will be highly personalized and individualistic. And that's going to rely on things like the right data and the ability to get genomic data and the ability to have very powerful computers look at that data and target individualized treatments. An interesting little stat you may not know, sequencing each person means creating a little mini database of about 3.2 gigabytes. Probably doesn't fit on your flash drive yet, but it will in the future. And that means that we need modern data analytics and technology to be able to push through that data and understand what are the cures and the treatments, and the insights that are capable of pinpointing those cures. I think all of that will require some changes in government policy, on both regulations and in laws and legislation. It requires, I think, a large investment in data and infrastructure that perhaps the current Congress has not yet been willing to make, but which we are willing to advocate for. I think it also requires changes to our, what I would say is our outdated privacy laws, and recognizing that things have changed and evolved over the last 20 years since HIPAA was first enacted. I think it also requires sharing of information across systems and providers, the so-called holy grail of interoperability that many of us are seeking, to unlock that data at the clinical level, at the EHR level, combine it with claims data and real-world data in order to be able to do some important throughput. So my opinion is personally that we need to do a better job of informing Congress and the administration and the public generally about why this is important and what we might do collectively as advocates. The important thing, though, is that private efforts can start without delay. Right? We can go out today and start doing stuff. As a private sector, we don't need to lobby Congress. And there are important companies and individuals in this room who are doing just that. So one thing, does anyone wear a Fitbit here? Raise your hand. I always got one. A couple of people, jawbone, anyone? Thank you for being a data donor. Maybe one of the private sector efforts, just like when you sign up for a driver's license to become an organ donor, you could sign up to become a data donor. I know it sounds like a goofy idea, but that kind of thinking, I think we should start applying to all aspects of our lives so that we can start leveraging data to improve health outcomes and lower costs in America. So those are just some initial thoughts. Our conference goals are threefold today. The first is to explore those political, legal, and operational issues that are holding us back. And the second issue is to identify the necessary reforms so we can speed the translation of data into knowledge, products, and ultimately cures. As a result of this conference, shortly after this conference, we're going to be producing a white paper that will become ultimately an advocacy roadmap for us to use in 2015 and beyond. So that's why I say this is not a dog and pony show. We're going to produce something from this conference. We call on you to be partners in that process to help us produce that advocacy roadmap. And then ultimately join us in our efforts. It will be a collaborative process. So those three things that we're going to hope to get from today is going to be assisted by 3 panels with really exciting panelists and experts from across the country. We're also going to have presentations from a number of folks that will help us in this task as well. And so without kind of further ado here, it's my personal privilege to introduce Dr. David Ledbetter, who's the Executive Vice President and Chief Scientific Officer at Ambassador Health System. You can read about his biography in your booklet. I'm excited because I spent a fair amount of time and effort when I was congressional staff trying to get new resources for autism. And Dr. Ledbetter has spent a considerable amount of time and effort on discovering the underlying etiology of developmental disorders, including autism. He's done a lot of other interesting things, but I personally want to thank you for your work, and we're pleased that you're joining us for this event. Thank you.
Thank you, Scott, very much, and thanks to you and to Alice Borelli particularly for the invitation to kick off this meeting this morning. I wish I'd known about your interest in autism. It would have been much easier for me to present my normal lecture on the genetics of autism. I'm trying to do a little bit broader commentary today from a large integrated healthcare system perspective on the future promise of genomics in a learning health system environment. I did want to comment on your notion of data donor, which is related to a campaign I'm trying to get off the ground at Geisinger. As a geneticist, we don't just think about the implications of our research on society as a whole, but to our families, and that means to our children and grandchildren. And as a geneticist, I would make the statement that the greatest gift that you could give to your children or grandchildren is your genome sequence and your health records. That's the most valuable thing that you could give to your children or grandchildren, and it doesn't cost you anything. A little bit different version of your data donor notion. I hope everybody in the room has at least heard of Geisinger. Not everybody knows a lot about it or where we are. This map shows you our patient population coverage. Some of you may remember James Carver's famous description of Pennsylvania, which is Philadelphia to the east, Pittsburgh to the west, with Alabama in the middle. We serve all of the Alabama portion of Pennsylvania, with headquarters in Danville, Pennsylvania. We're an integrated healthcare delivery system, which means on the left we have physicians and other healthcare providers. In the middle, we have tertiary quaternary hospitals and a large number of primary care clinics distributed through a 40-plus county area. And we also have an insurance arm, the Geisinger Health Plan. This means, the implications for this conference is we have comprehensive data from the payer and provider side about a large percentage of our patients. About a third of the patients that we provide clinical care to also have Geisinger insurance. So we have claims data as well as clinical data on that third. Glenn Steele, who left academia to move to Geisinger Health System almost 15 years ago, makes our physicians nervous and our board members nervous when he refers to Geisinger not as a healthcare delivery system, but as his laboratory. So he comes from a strong academic research background. He views the integrated nature of the system as a closed model system that allows you to measure the outcomes of any technology, device, care process, re-engineering experiment. And also because you are the provider and the payer to measure the impact on total cost of care more accurately than just about anybody. So I've modified the typical slide showing the structure of Geisinger to indicate Glenn not as the CEO and president, but as the principal investigator of this laboratory, with the mission to conduct experiments in re-engineering healthcare to improve patient outcomes and reduce cost of care. Part of the reason he was attracted to this environment was that Geisinger was a very early adopter of the electronic medical record. Depending on the version of the story you believe, we were Epic's second or third customer in 1995, and some of the other attributes are listed there. They were wise enough to create an enterprise data warehouse that would make this data more accessible for research as well as for operational and quality purposes. This was initiated in 2004, so 10 years ago. So this has EHR data, financial data, operational data, claims data, etc., in a much more searchable, accessible form than would otherwise be. So where does genomics fit into this? I'll start just talking about my own evolution in genomics research, which I think has paralleled some of the interest in the NIH National Human Genome Research Institute, one of the 27 or 28 institutes at NIH. Eric Green published this figure showing the 5 domains of genomics research that were of interest to his institute, starting with basic science on the left, moving to translational and clinical research, going to the right. And then the last one, improving the effectiveness of healthcare. Who would have thought 10 years or so ago that the National Human Genome Research Institute would be interested in implementation sciences and actively involved in moving the technology and information from genomics into real-world healthcare settings. So Geisinger views itself as participating in some of the translational research, but being an ideal laboratory for the implementation science to determine the clinical utility of this new technology and data capability. And as a healthcare system, of course, we care about value, and that is improving outcomes, but paying attention to the cost as we're doing that. In 2009, Geisinger did a year-long strategic planning process around research and updated their mission that has always included a strong commitment to innovation and research. And the overall theme coming out of that strategic planning process was personalized healthcare, similar to many places, with an emphasis on genomics. It was a result of that strategic planning that they made an increased commitment to research, including the creation of a chief scientific officer position at the executive level, which I was recruited into in 2010. So what we're interested in is determining the value of genomics over the lifespan. We have a unique population that's relatively stable, so we can think about investing in preventive health measures that won't pay us back for 10, 20, 30 years or longer. And so if you view it that way, then the cost of genomics and the economic modeling and the ROI can be done over a much longer term scale than in other fragmented delivery or reimbursement systems. And obviously with genomics, in theory, you only need to do it once to do the DNA sequencing of an entire genome. And this leads to important benefits in pharmacogenomics and in many cases for patients with undiagnosed disorders, which I'll mention again later. It raises a lot of questions, and that's the main focus of our research, are around how do we do this, and how do we store the massive amounts of data that are going to be generated, how do we make it available to physicians and patients wherever they may be in our system, or if they move to other systems. How do we generate the data on clinical utility and benefit to the patient and the impact on total cost of care and other issues. So I thought since I was first this morning, I'll make a few comments on where we are today with genome sequencing. So-called next generation sequencing is just a technology platform description. For whole exome sequencing, which is not the whole genome, but the genes, so it's only a few percent of the entire genome, so it's currently cheaper, costs roughly $2,000 to $3,000 to do whole exome sequencing today in a clinical laboratory setting. Although for an undiagnosed patient, particularly children, it's more informative if you study the parents and the child. So 3 times $2,000 to $3,000 is $7,000 to $9,000 to do a family trio to get the maximum informativeness. So still not cheap, but I think some key papers came out in a special issue of JAMA on November 12th, so just a month ago, that are very useful references and amazingly consistent in their finding. The first one from the Baylor College of Medicine Genetics Diagnostic Lab on 2,000 clinical diagnostic cases with a 25% diagnostic yield from whole exome sequencing. This is in many cases after many other genetic tests were already done and were uninformative, and now whole exome sequencing solved the problem, stopped the diagnostic odyssey, stopped the need for other diagnostic medical tests and clinical evaluations, just determine the cause of the patient's clinical presentation. Another group from UCLA did a little bit smaller study, but came up with an identical 25% yield. So we have very good data now on the clinical validity of whole exome sequencing. Clinical utility, we still need more data in terms of what do the providers and the families do with that data, what are the actionable items. There's a little bit of data suggesting that it does alter management and treatment in a small percentage of patients. One recent study suggested almost 50% of the time physicians report that it altered their management and treatment of patients, but we need much more clinical utility data if we hope to move this into routine practice with good reimbursement. So what about whole genome sequencing? Whole genome sequencing, this can be available on a clinical basis. This is not as popular as whole exome sequencing yet because of the cost. This is my whole genome sequence on a 500 gig hard drive. Illumina will do whole genome sequencing in a clinical laboratory setting It's $5,000 typically out of pocket, $500 to get the raw data on a hard drive. It takes up about 100 gigabytes compressed files for one person's genome sequence. This is my clinical report, which came to my primary care physician, Dr. Rolston, who's the head of general internal medicine at Geisinger. He was pleased to order this test on my behalf. I met with him when the clinical report came back, and I had to explain the report to him, which is one of the obstacles that we face with this technology. Is most, even really good internal medicine and other physicians won't know what to do with this information, so you need a professional geneticist to help with the interpretation. Although he was quite pleased with the understandability of the report as I walked him through it. In the clinical report, I get back some variant statistics. So of this 100 gigabytes of raw data, there are about 4 million variants or differences between my genome and the reference average normal human genome. So there are 4 million candidate variations that could have some impact on health and disease. So one of the issues we have to learn in the future is what data do we need to store long term? The entire raw data files, which are big, the variant files, which are much smaller. Over time, we'll learn which of those variants have any clinical relevance and which don't. This is a distribution of the sequencing depth. So when you're doing whole genome sequencing and exome sequencing, you sequence many times in order to improve the quality of the base calls. Illumina, on a clinical basis, will make a call if a particular base is covered, high quality sequenced 35 times or more. And this is what generates the 100 to 150 gigabytes of raw data. Here's the results page, and the first category is called clinically significant findings, and you can't read it from there, so I'll read the highlights. No pathogenic or likely pathogenic variants were found in the 1,691 genes evaluated, so we think we have 19,000 genes, but we don't know what all of those genes do yet. So Illumina at the moment has determined that 1,961 have sufficient evidence in the literature to make a clinical evaluation and recommendation to physician. Then they give a carrier status report. We have predicted for a long time all normal humans probably carry around 5 mutations for autosomal recessive disease, so you're not affected, but if you have a child with another carrier, you have a 25% risk, and I had 4 of these, including carrier for a cystic fibrosis mutation. And then you get a detailed pharmacogenomics report, which can be very informative. Again, mine was pretty vanilla in that most of them were intermediate metabolizers for most of the common drugs that show up on this pharmacogenomics list. So coming back to Geisinger and what we're doing, we have quite a number of activities in genomics that all fit into this notion of implementation sciences, figuring out when is this useful, how is this useful. We participate in a number of NIH-funded and PCORI-funded network collaborative projects. And then more recently, we have a major large-scale sequencing project with a pharmaceutical partner, Regeneron Pharmaceutical in New York. We started a biobank in 2006 that we call MyCode. In that system, which I'll show you some data in a minute, is over 60,000 consented patients and accompanying samples. We have a system where we usually don't draw a special research blood sample. We put an order into Epic. The next time the patient has a clinical blood draw, they automatically take an extra tube that goes to the Genomics biobank that actually works. So it costs almost nothing and doesn't require an additional sample on the part of the patient. This slide is outdated, and I updated this, but somehow put the wrong version onto this laptop. So we're over 60,000 patients in a combination of primary care, so some healthy individuals, as well as specialty clinic environments. And the typical list of common disorders of adulthood in the United States, and Pennsylvania has at least its fair share of obesity, diabetes, etc. Just to give you a flavor, of course, we're concerned about privacy and security of the data. So for a long time, our enterprise data warehouse has had a data broker system with a firewall between the personal health information, PHI, and the research investigator, so they don't have direct access, but access the data through a data broker. We participate in a national network called the eMERGE network for electronic medical records and genomic information, sponsored by the National Human Genome Research Institute. This shows you the participants. It's now 9 different institutions that combine their electronic medical record data and their genomics data. This is very powerful in terms of doing genome-wide association studies. Has been a very productive collaboration, and is one example of the current state-of-the-art, which can be a little clunky in terms of data sharing, both on the clinical side and on the genomics data side. We've recruited and built a large genomics team at Geisinger, including physician geneticists, as well as a large number of genetic counselors who will be involved in direct patient education and counseling around this information. We participate in a new project funded last year, again by the National Human Genome Research Institute. It's a data sharing and data curation project referred to as the ClinGen, or Clinical Genome Resource project. Again, a number of institutions, and its goal is to create a single source that's clinician friendly to look up whether a genetic variant you find in your patient has ever been seen before, and is it disease associated or it's common in the general healthy population, right? So right now, that's not easy to sort out even for a professional molecular genetics diagnostic lab or professional genetics clinic. The goal of this project is to have expert committees curate the world literature and to create a single clinician-friendly resource to find all of that information. Now I'm going to come back to our own MyCode, and we've expanded the name of this to a community health initiative to emphasize the patient and community engagement part of this. One of the unique things of Geisinger is the very positive relationship between the healthcare organization and our community. And we recently decided, or a little over a year ago, decided we needed to update our genomics consent form around our new goals and project. Partly this was related to an important national discussion around the duty to return results that come out of research projects that have clinical significance. So instead of this complete separation of research and clinical care, particularly for us as a healthcare delivery system, all of our research volunteers are our patients and their families. So we feel a strong obligation and duty to design our research in a way that allows us to provide back any clinically useful information to the individuals and families who have volunteered to participate. Cost of sequencing was going down, other reasons. So we did a series of focus groups and tried to sample some different geographic areas. And we talked to them about the different types of genomics information that we might find, and asked them, would you want us to give this information back to you? We talked about pharmacogenomics information. We talked about being a carrier for an autosomal recessive disorder. We talked about autosomal dominant disorders that have a very high risk, high penetrance for a disease that might be preventable or treatable, and also for a very high-risk dominant disorder where there was currently no treatment and no way to prevent the disease. And did they want us to tell them about genetic changes we saw that we don't know what the heck they mean today? Have no evidence that they're clinically relevant or not. Surprisingly, and I'm not sure we believe this completely, they said they wanted all of the results. But then when we talked to them further, they said, we trust you as the medical genetics and genomics experts to decide which information is clinically relevant to me and my family. After all, you are my healthcare provider, so we trust you to do that. But we do want you to return the clinically relevant information to us and to our doctor, preferably at the same time. We don't want to always only hear it after our doctor hears it. We want to know about it simultaneously. And that we trust and want you to develop educational materials to help us understand this. And we want you to put the information into our electronic health record so that anywhere we are in the healthcare system, people will know about that genomic information. So we went through a process after these focus groups and a lot of discussion and consultation with both a bioethics advisory committee, including local lay representatives from our community and from our patient population. And the updated consent form for our genomics work said we're going to do genomic analysis up to and including whole genome sequencing. We intend to return the clinically relevant, medically actionable results after confirmation in a clinical, CLIA-certified laboratory. And we're not going to return results at the moment that are not known to be medically actionable. And our experts are going to decide that. We also said we're going to share data with other researchers, and your data might be used in partnerships with for-profit pharmaceutical companies for drug development, other commercial applications, and you won't receive any financial reimbursement for that commercial activity. So this went through our IRB last fall. We're now reconsenting all of our original participants in the MyCode project and implementing this on a prospective basis with essentially no measurable difference in the consent rate by our patients, which is typically over 90% in every clinic and most often over 95% in the great majority of clinic settings and patient population. So now I had recruited all the experts, built the infrastructure, waiting for NIH to announce a request for applications for large-scale population sequencing, and it didn't happen. So we found out that Regeneron Pharmaceuticals, which for many years had been an expert in mouse genetics, functional genomics research, was now interested in human genetics and was shopping for healthcare delivery partners. So we worked with them, and in January of this year announced a long-term partnership with them to sequence at least 100,000 patients in the Geisinger system. This was a great partnership because of our complementary expertise. We have this very unique community, very stable population, 3-generation families, perfect for genetics research. They had a very strong scientific team. They're a successful pharmaceutical company and cash rich, so they could build a genome sequencing facility, state-of-the-art, with as large a capacity as any sequencing facility on the planet. They did that de novo at the beginning of this calendar year, and they're now sequencing 1,000 patients per week from our healthcare system. So our goals on this study are to identify disease risks and prevention based on each individual's genetic information, figure out how we can use that information to individualize, personalize, improve treatment, optimize outcomes, identify the right drug, right dose, etc. Improve the health of Geisinger patients, and then patients, from what we learn, disseminate that information nationally and globally. As a large healthcare system, we're as paranoid about security and privacy as any. So our IT group and our security ISO group is extremely conservative and careful. We have a lot of experience. So everything that we do involves our IT data security experts locally and beyond. So nobody can promise absolute privacy and data security, but we work hard at it. Now, in discussions with our patients, we quickly realize there's already data available that if you sequence perfectly healthy adults, we know that about 2% of the time we're going to find a mutation in a gene that's an autosomal dominant gene with a very high penetrance, meaning you could be at very high risk of cardiovascular disease or cancer, and you may not know it. So if you don't have a big family or don't know that family members have had particular cardiovascular or cancer risk, you may not have been referred for genetic family history, pedigree analysis, and other medical testing. So we're going to discover genetic mutations in currently healthy individuals, and we know that's going to be 1 out of every 50 people. So we view that as a benefit to the individuals who have volunteered to participate in our study. We also know when we find that mutation in the research participant, since they're dominant genetic diseases, if we then start evaluating their close relatives and work our way through their family, we'll find 6 more individuals for every case. So that means we're going to find a lot of people who don't know they're at high risk for cardiovascular disease, cancer, and some others. So as a result of this collaboration, we've greatly increased our recruitment and consenting. Recently got through our IRB. Online recruitment and consenting online, because face-to-face consenting, although we have a very high success rate, is time-consuming and expensive, and that's been our biggest bottleneck. And also has enabled us to greatly expand our data analytic core to be able to compare the genomic data to the clinical data. This is an older version. So this allows us to now start thinking about our research, not as starting with different clinical cohorts defined by their clinical presentations or phenotype, but the starting point for research is you know an individual has a mutation in a gene that puts them at very high risk for a particular disease, or that they have a pharmacogenomics profile of a certain type. We're referring to that as a genome-first approach to research, and we think this will transition into clinical care. So in the first 100,000 patients, as I said, we know that we're going to find a DNA sequence alteration or mutation that may indicate a life-threatening condition unknown. So how do we choose those genes? Well, the American College of Medical Genetics and Genomics is the national professional society for the field of medical genetics. They've had an expert committee discuss this and say, let's be very conservative and only take the genes where we know a lot about the gene, the disease it causes, and that if you have a mutation in this gene, the odds of developing this disease, usually at a younger age than normal, is extremely high, so very high penetrance. They recommended communicating those results when they show up as a secondary finding, doing exome sequencing for other clinical purposes, for 56 genes, only 56 out of the predicted 19 or 20,000 genes. We've expanded that a little bit by adding similar genes that cause the same phenotypes and meet all of the criteria to 76. And again, this is the criteria for the American College, and 2 faculty members in genomics at Geisinger were on this original national professional advisory committee, and Dr. Krista Martin at Geisinger chairs this committee currently. So these are some calculations of what we're going to find in our genome sequencing project. We know that for breast cancer high-risk genes, BRCA1 and 2. The population prevalence incidence is about 1 in 1,000. Lynch syndrome, cancer, high cancer risk syndrome, 1 in 1,500, and you see some of the other hypertrophic cardiomyopathies and hypercholesterolemia. So we can do the prediction that we're going to find about 2,000 individuals with a mutation. Then when we add their 6 at-risk relatives, another 12,000, we'll find 14,000 people from the starting population of 100,000. Only 2,000 are the research participants themselves, but their family members are likely to live in our catchment area and be Geisinger patients as well. So this number might be a little high, but not too far off. So we're hiring a lot of genetic counselors in anticipation of returning these results and trying to figure out the most efficient way to educate patients and educate our physicians and other healthcare providers. So we're going to learn a lot from this experience. We do have an outstanding director of bioethics who provides clinical bioethics consultation for our healthcare system, but also is a researcher previously at Georgetown University and the National Institutes of Health, Dan Davis, who has a great external advisory board, bioethics, chaired by Kevin Fitzgerald at Georgetown, but including bioethics experts in genetics and also patient and community representatives as well, to make sure that as we explore how to do this research and then how to implement this new information in a healthcare setting, we do it in an appropriate ethical framework. Geisinger was a participant in a project initiated at Beth Israel Deaconess in the Boston area, in collaboration with a few other groups, called Open Notes, which provides patients access to their physician notes. This was so successful in the pilot phase, which has been published, that we've now rolled this out to every clinical area at Geisinger, meaning all patient physician notes and other patient records, including lab records, imaging records, imaging data, are available to patients. The only exception at the moment is psychiatry, where we haven't figured out exactly how to present that information in a totally open fashion. So our full intention is to figure out how to make raw sequence data, variant call files available to patients if they want. So this meeting today is not the only group talking about these issues. Just Monday of this week, I was supposed to be at but couldn't attend an IOM meeting. I know some of you were at this meeting. I spoke to Lynn Etheridge before his presentation about genomics-enabled rapid learning system. We see ourselves as a learning health system with a primary focus on personalized medicine using genomics information and tools. So this model is very relevant to what we're trying to do. And in his talk, talked about current national efforts by a variety of important agencies, NIH, FDA, PCORI, to talk about common data models, including now genomics data information, and upgrading the existing model of rapid learning health systems to include genomics data and the issues around genomics data storage, transfer, etc. And encouraging this national interest, national coordination among different groups. So this vision statement comes from an informal talk I gave a few weeks ago at Illumina workshop, discussing many of the same issues about whole genome sequencing and what it's going to take to implement it on a routine basis. This is my personal vision statement, not reflecting Geisinger or anybody else. My goal in moving to Geisinger was to learn how to use this information appropriately towards a goal of universal genome sequencing, preferably at birth, as part of newborn screening, so part of a public health and medical project. Obviously, we need to learn a lot, and costs need to come down significantly. And in doing this, we need to think about this primary stakeholders, the patients, their families, providers, payers, and employers. And most large employers are self-insured. So during thinking about reimbursement issues and who needs to help generate the clinical utility data and who do we need to convince that this will improve health, well-being, and productivity, employers are an important component. So for newborn screening, are people interested in it? Well, Robert Green at Harvard just published this paper in the last week. That was a survey type study of parents who had just delivered a baby. So in the immediate postpartum period, surveys were done to say, would you be interested in genomic information on your newborn baby? And the great majority said yes. So this is a nice example of not presuming what patients may or may not want, but just ask them and let their response guide your research and clinical implementation. Now again, thinking about this meeting and the implication of genomics data in terms of big data challenges, I've been working with our IT group and clinical data group to try to get some idea how big is genomics data compared to current EHR data. Maybe other people in the room who know this and can speak to it later, because I'm a novice in this area. So I said, my genome is 150 gig raw data, 10 gig variant file. If I put that in my electronic medical record, is that a lot or little? How much data is there? So they did some preliminary studies looking at the number of visits, encounters with the healthcare system by age, because I was curious what over the lifespan, what the number of encounters and how much EHR data, clinical data there was. And so this graph is broken in the first couple of bars into 5-year intervals. So in the first 5 years, there's a lot, goes down, and then starts coming back out and peaks later. Oh, this is a cumulative plot here. No, I'm sorry, it's not cumulative. It's by 5-year intervals to the left and then transitions into 10-year intervals after that. So if you make that a cumulative plot of the average amount of patient data, and this is in megabytes of data in the electronic medical data, you get to an average of about 20 megabytes, so not really a big data on an individual patient basis. So now I've just added, what if we did newborn screening and we started with whole genome sequence data at birth on an individual in their electronic medical records, so now you dump in 150 gigabytes or 10 gigabytes of genome sequence data. The cool thing to me is to now think about the clinical research opportunity as that person goes through the healthcare system and you acquire additional clinical data and also real-world data, you can capture that, and look back at the genome sequence data, look for correlations and do discovery research against this existing genome sequence data. So this is very preliminary thoughts, and I'll be curious to talk to some other experts here about how to think about the impact on our EHR clinical data if we could add whole genome sequence data. We already do newborn screening by tandem mass spec. We only look at 30 diseases as part of our public health newborn screening program. We throw away all the other metabolomic data and don't do anything with it. So you could have complete genomics and metabolomic data on every baby born and then look at the health implications after that. So to summarize, we are a high-value integrated healthcare system that I think provides the ideal genomics learning healthcare laboratory to perform experiments to improve patient outcomes and reduce total cost of care. Genomics data available from birth, perhaps prenatally in some cases, may allow personalized health guidance in the form of prevention and optimized treatment. The lessons learned, I realize we have a lot of unique attributes that make it ideal as a laboratory to do these pilot studies. Not everything will be generalizable outside of our unique population and structure, but we expect that many of the lessons will have some general usefulness. But there are a lot of gaps in what we know, and that'll be parts of the topics here. I think the biggest is clinical utility, so costs will come down, but we never, the researchers in genetics and other areas of technology are excited about the technology, excited about the clinical validity, the sensitivity, specificity, but we don't get to the clinical utility. So does it really change outcomes? How does it impact total cost of care? How does it fit in a value-based healthcare system? The other big technical problem right now is we're not yet at a point where we have a single whole genome testing technology and platform. So we're doing exome or genome sequencing after we've done whole genome microarray or copy number variants. So we're still doing multiple different tests. So we need that to all converge on one comprehensive genetic test that becomes affordable. So there's still some work to do on the technical side. So I think that's my last slide. So I'll stop there. Sorry for being a few minutes over. I don't know if you want questions now or just wait for the panel.
No, if folks have questions, by all means. Right over here.
Yeah, I was just curious, that 2% figure for the 100,000, that was exome sequencing, just to remind me.
That's exome sequencing.
So would you anticipate the figure to go up with full genomic sequencing?
So the number of variants we already know is much higher. The problem with whole genome is most of those variants we don't know what they do yet. So we need to figure out which ones are clinically relevant. So by definition, they're mostly not within genes. So yes, it will go up. It won't go up rapidly if we just switch from exome to genome sequencing because we don't know the significance of most of the variants.
And the size you were making of the databases, that did not include imaging.
It did not include imaging. I meant to say that. You're right. Exclusive clinical data exclusive of imaging. Josh?
Yeah, so I have a question about the 76 genes that you're returning to patients. Do you anticipate facing some new dilemmas about what to do about those results? Do you feel like there's good guidelines in, let's say, the absence of a phenotype about what to do about cancer risk findings? Or are you going to have to develop those as you go along?
Yeah, so I mean, it introduces some uncertainty about penetrance because penetrance comes from clinical cohorts and not from healthy populations. My personal opinion is that penetrance may drop a little bit. Everything that we know about breast cancer, for example, suggests it's not going to drop very much. Right? So when we give the risk figures, we may modify it and not give just the published high estimate of the penetrance, but give a range of penetrance based on that level of uncertainty. But everything in the original 56 and the 20 we've added are already known to be 80% penetrant and above, so very high risk, lots of data. It's not going to change that much.
Quick question. You mentioned the Open Notes initiative.
Yeah.
I'm curious, are patients able to download the information, and if so, what are they able or allowed to do with it?
So I don't think at the moment they're able to download it, but I'm not sure. It's a good question. Our intention, just like with Illumina, is to be in a situation where patients want, just like today they get medical records and they pay to Xerox for the fee to Xerox. We'll figure out mechanisms at cost to provide large amounts of data on hard drives or other mechanisms, or explore ways that we could allow them to download things.
And, sorry, just to follow up, contractually you would let them do anything? Meaning they wanted to upload it and share it, that would be up to them?
So, again, I'm not speaking for the entire institution because we're not there yet. My personal view is it's their data, they can have it, they can do whatever they want. And we need to be prepared to deal with the follow-up questions and issues that come up from that.
I actually wanted to just comment on that. So probably there are bigger experts in the room than me, but HIPAA gives folks a right to access to their data and their medical record. And that once it's theirs to do with that copy with whatever they want. So that's actually a provision in HIPAA, and there have been big penalties for providers that don't support
And
that.
there's recent federal legislation that laboratory results and data all,
Correct, you can
must
get
be
direct
provided.
access to your lab results, and you also have a right to an electronic copy of any information that's maintained electronically, as long as you're asking for it in a manner that can be provided. And also just a reminder that under meaningful use, patients can download coded data or share that as CCDA, which is the a summary of the clinical conduct. Things.
Thank you, Claudia.
Remiss.
Thank you, Dr. Lindberg.
That said, the purpose of this panel is to talk about the promise of genomics, and we're really going to try to do this as a round table. The panelists, all the way to the far right, or at least my far right, your left, moving over here, are Peter Hodge, who for the purpose of this conversation, I don't know what he does professionally, but he is the parent of 2 children with cystic fibrosis and has been very active in the Cystic Fibrosis Foundation. To his left is Dr. Josh Peterson, who is a primary care physician and assistant professor of biomedical informatics at Vanderbilt University. And to immediately right is Dr. Toby Bloom, who is the Deputy Scientific Director for Informatics at the New York Genome Center, and she'd been at the Brand Institute doing something similar for the previous 10 years before that, PhD in computer science from MIT. So we had a conversation, as you always do on one of these roundtable conversations. I don't know who I was going to start with first, and they still don't know at this moment, but the journalist in me says, let's go with the anecdote. So Peter, I'm going to ask you the first question, which is, you know, you have 2 children with cystic fibrosis. You know, when you think about genomics, the Human Genome Project, Dr. Francis Conlon, he identified the gene. That's what he was actually renowned for. Now head of NIH, he run the Human Genome Project. Just give us a sort of quick background of what it meant, you know, the genomics and how it affected your family and the treatment of your family.
Yeah, sure, absolutely. So, good morning, everybody. My oldest daughter, Olivia, was born 22 years ago and was diagnosed at birth with cystic fibrosis. If you're not familiar with the disease, it's a fatal disease that impacts the lungs and digestive system. At the time when she was born 22 years ago, life expectancy was about 18, so it's a pretty hard diagnosis. Just 3 years before that, so 25 years ago now, Francis Collins and a team from Sick Children's in Toronto had actually discovered the cystic fibrosis gene. It was the cover of Science magazine, and it was really the first genetic disease to be isolated for the gene. So about 3 years later, my daughter's born, diagnosed with a sweat test, which is a diagnosis test for it. And then she had her genetics done. So 22 years ago, we did a genetics on the CF gene. At the time, they could only look for one of the specific mutations, the most common mutations carried by about 80% of people with CF. And sure enough, she had one of those. They couldn't find her other mutation. Now we know there's at least 2,500 different cystic fibrosis mutations. So it's been a long, hard road over the last 22 years, but just 2 years ago, we realized the promise of genomics when Vertex Pharmaceuticals got approval for a miraculous new drug, which is really the first drug to treat individualized medicine against a specific mutation type for a disease. In fact, when I meet somebody else who has children with cystic fibrosis, the second question we'll ask them is, what mutation do you have? Which is really kind of strange to live in a world where patients want to know that information about each other.
Okay, so let me go to you, Dr. Peterson. What is the clinical relevance of, a lot of the information that we were told about in the first presentation? This is a very specific example, cystic fibrosis, but now we're talking about 14,000 possible out of 100,000 patients being identified that have some kind of gene variant that might have some kind of clinical significance. But what is the real clinical significance of most of the genomic information that's out there?
Well, I think the list that David presented, the significance is very high. I mean, that's one of the reasons that they are returning those results, is that there are potentially actionable items
that the clinician can take with the patient in response to having that variant. So that might change the way that you screen that patient for cancer over time. It might change how you counsel them about symptoms that they may encounter through their lifetime. And certainly for part of that list, which is related to pharmacogenomics, it may be immediately pertinent to the way that you treat those medications. That list of variants that affect your medications has grown tremendously in the last 10 years. I think about 10 years ago, there was only 5 that were listed within the drug labels at the FDA, and now there's 150. So very quickly, we're developing a knowledge base of how to tailor treatments using genetic variants, and that is oftentimes the most immediately actionable part of that of the whole genome sequence information, although a lot of the other information is very important.
Okay. Dr. Bloom, I want to get you in the conversation as well. So why don't you talk to us about how the research that goes on that Dr. Peterson just referred to. So in other words, they're identifying variants that actually do have an impact on clinical decision making. How is the research done so that they can be identified that way?
So that's an interesting question. The New York Genome Center, I don't know how many of you know this, it was formed as a collaboration of 12 large medical institutions in New York. So we were formed with the explicit mission of bringing genomic science to bear on improving healthcare and doing it now and quickly. And so I think one of the things that matters in research right now is we're obviously making great progress. We have Kaleido, we have drugs for cancer. So far, the low-hanging fruit has been the diseases that can be treated by treating one variant, one gene. The next big push has to be in looking at common diseases, things like type 2 diabetes, like autism, schizophrenia, autoimmune diseases even. These are all diseases that are probably caused by interactions among multiple genomic variants, but also interactions with the environment, exposures to infectious agents sometime in your lifetime. The complexity of doing that is still, we're still starting to tackle that. There was a research paper that came out recently on type 2 diabetes, not by us, by the Broad and a collaboration with the Broad actually, on type 2 diabetes that I believe took 250,000 genomes to be able to validate the mutation. So we're clearly in the big data space here. And when we need to add in clinical data, deep clinical data, to find medical histories and figure out how that relates, and other things. Here's my jawbone, folks. Yes, I wear one. We are actually using that in some research studies now and correlating it with genetic information. This really brings us to a point where, in order to make progress, we are going to have we're going to need unprecedented collaboration between all the stakeholders, the patients and the patient advocates, the regulators, the physicians, the scientists. Geisinger, I think, has this real advantage in having a somewhat closed system. Once we get to trying to pull that data together, to be able to share data across systems, and it's critical that we be able to share data across systems, to being able to get clinical data that we can pair with genomic data, and protect patient privacy and understand what the privacy issues really are. We're all going to have to come together to do that. And I think all I'd say here is, can we figure out how to do that? Can we all figure out here what the right way is to collaborate on that?
I want to go back to one thing, though, that you just said in your talk and get the others to address this. and that is just, there's low-hanging fruit of one gene, one variant, and now we have this sort of tidal wave of genomic information. I mean, it's still small relative to the whole patient population, but now we're getting hundreds of thousands of people who are having their genome sequenced. And you get, I'm sure that there must be quite difficulties. There must be a lot of difficulties in sort of deriving real actionable information out of that. So I'd like to sort of give us where we're at in terms of really being able to understand that some of the more complex questions of a pattern of variation that actually becomes actionable information in a clinical sense.
Are you asking me first or
Dr.
everybody
Peterson, I think
first?
maybe,
Yeah, I think you bring up a really good point because, you know, genetics is rarely deterministic, particularly once you get beyond, you know, sort of a low-hanging fruit. There's a few things that are highly penetrant, so to speak, and so those things, if you see the variant, you can predict reasonably well what's going to happen. But for most genetic effects, they're combined with environmental effects, of course, to give you the disease, the ultimate disease or ultimate phenotype. And so for clinicians, one of the biggest challenge is how to combine that genetic information with other things they know about the patient. So I'll give you a specific example. We return to our cardiologists information about essentially a blood thinner type drug or antiplatelet drug, clopidogrel, about whether it's efficacious or not. And when some of the genetic pattern sort of says, well, this is going to be more efficacious than you intend to be, and so it might influence bleeding, and some of the genetic pattern says, you know, it's going to be less efficacious. Well, they have to sort of take that genetic information and combine it with things like, well, this patient has already bled, or this patient had a stroke in the last 6 weeks, and so they're at risk for bleeding in their head, or this patient is on another drug that influences bleeding. And to put all that together in a quantitative sense, we're not quite there yet. We don't have all that. We don't essentially have the calculation to say, well, we think the patient is predicted to bleed or not predicted to bleed. So that's the kind of thing they're having to do in their heads, and that's a major challenge for us. We have to sort of help them get through that decision-making process.
Wait, look, just to follow up on that before I go to you, Peter. So if I'm a clinician and I'm getting a kind of mixed message, how am I supposed to respond to that? I mean, think about all the doctors out there who are making that decision about clopidogrel. That's a very commonly used drug, right? And now they're being told, well, maybe yes, maybe no. I mean, it seems to me that it could be a very difficult decision-making process for the physician in the early stages of this research.
I think there is some frustration, but it depends a little bit on whether this is a kind of physician who's an early adopter or someone who is not ready to move into making these kind of decisions. But also recognize that the physicians are generally used to making decisions with some uncertainty, and they're also used to making some decisions using multiple types of data. So you do get, I think, a very rational, reasonable response most of the time to getting genetic data that gives you a higher risk of an event, but it's not going to tell you for sure whether it's going to happen.
Peter, you mentioned about the multiple variations we now know in cystic fibrosis. This is a fascinating part of this question. So when we find one gene, the low-hanging fruit, it turns out, has a lot of different subspecies of the low-hanging fruit. So what does it mean in a research sense then? And from a patient's perspective, does that mean people are actually failing on some of the new early therapies and therefore become target populations for new modifications or follow-on therapies?
Yeah, so there's a couple pieces to that. One, I think, is that we think about a disease like cystic fibrosis, or maybe any disease, and rarely do they follow a course that you can actually map out. So a doctor might say, okay, here's what I think is going to happen, but it rarely does. So, for example, with my two daughters, one has really bad lung disease, the other has really bad liver disease, but actually pretty good lungs. But they both have the same genetics, right? Clearly there's something else going on. So the idea to be able to back into the big data and say, all right, here's cystic fibrosis, but what are the modifier genes within here that we can relate the clinical features, what's actually happening, and look back and draw out big information. And that's a perfect example of the ability to use big data. And then I think apply all the other stuff we've got to add as well, environmental and behavioral information to that as well. So I think that's one aspect of big data applied to a specific disease here. To the second part of your question about do these therapies work on different variations of mutations? Yeah, absolutely. So if you look at Kaleido, it only works on 4% of the population of people with cystic fibrosis. In fact, there's sort of 5 classes of mutations in the disease, and what we're finding with these new drugs is it's not one drug, it's not 2 drugs, it's a combination of these small molecule drugs that are actually impacting. So the ability for patients and physicians to understand the complexities of the information, the disease, and then apply that to the treatment is also something that we're going to have to figure out as we move forward here. I know it's not a one-size-fits-all at all.
Go ahead. I was just going to jump in here. From a research perspective, there's sort of another interesting answer to that, which is we're often looking for variants that haven't been seen before. We're looking at diseases where we don't know what the variant is. And so we don't know whether we'll find it. We're not looking for something that's actionable in this patient. We're not doing a test for something we know we can find. We're looking for things we don't know yet. Sometimes those things can actually be actionable. Sometimes we can find a variant that's a cause of a disease, but biologically, it has a similar mode of action to some other variant that there is a drug for. And so it hasn't been tested, there's no clinical trial for that disease, but there might well be a drug on the market that could at least help, if not be a real treatment or cure. And so we don't know what we're looking for. We don't know ahead of time what's actionable. And we don't know ahead of time whether what we're going to find should be used in treatment or not. And that causes lots of problems in things like trying to figure out how to craft regulations to allow us to move forward and to save patients' lives.
I think if I just heard what you said correctly, it's sort of like the off-label use of drugs issue that has been going on in cancer therapy for many, many years, where we actually never collected information on the outcomes of the people who were treated off-label. And so I would actually like to hear people address that question, which is capturing the information of clinical responses to genomic information. Where are we at on that whole question?
I'll address it first. So I think the concept of a learning health system in this space is really important. The gold standard for figuring out whether something is beneficial or not is still the randomized controlled trial, but there are too many permutations in genetics to run trials on all the different patterns, and so we need to come up with a system that's going to let us look retrospectively and say, given a particular genetic pattern, given a particular disease phenotype, and given a particular treatment strategy, this is the most successful versus this is less successful. And so building that system across institutions is something that the IOMs are interested in, NHGRI is very interested in, but we're at the very early stage. We have contributed both genetic and phenotype information at our institution to emerge, and I think that's sort of an early experiment, but it's not complete. We have patients who we see for a year, we have patients we see for 20 years, and so the question is, how can we acquire all of the electronic medical record data on our patients that presumably most of it's in sort of Tennessee, maybe some neighboring states, but that's a very big challenge.
So I'm fascinated by this issue of running clinical trials, and the problem, it seems to me, is that the data that we collect, we can see variation. Say you're starting with a premise that we have a population where we've identified a variation, but everybody's had different environmental responses. If we're out of that realm of environmental experiences, so if we're out of the realm of that low-hanging fruit, we know a gene variation causes a very specific disease. And now we're looking at patterns and we know they're different. How do we capture that information in the context of clinical trials or even trying to figure out what the impact of a particular gene variation is?
So I'm not sure there's one answer, but I think one of the really interesting frontiers of getting better phenotype information is to get it directly from the patient. So I think engaging patients who are interested in donating their data, and that could be quantitative data from biometric devices like Jawbone, or it could be
that they're going to answer some structured questions through a patient portal. Those are ways to get the other information, like environmental effects, that you might need to sort of sort out whether, what combination of genetics and environment might lead to the disease, or what might affect your particular treatment strategy.
Peter, I was going to ask, in the cystic fibrosis community, has this been, accepted, or are people doing it?
It has. So, one of the biggest problems we have, especially when we start to talk about personalized medicine, is that there is only one of me, right? So if I want to do a clinical trial, I really got to move to an N=1 model. If you look at what the CF Foundation has done in conjunction with Vertex and the FDA, which has been an incredible partner in this as well, has really pushed the boundaries of clinical trials with an N=1 model. And some of the smaller trials on some of these very small variants where you have maybe 20 people in the United States with that particular gene mutation, it had to move to this model, and it's actually been very successful. It's still too slow from a parent perspective, right? It takes too much time, it's too slow. I know we've got to do it, but I think it's a big issue around this question of how do we have the mechanisms available to approve all of these drugs in a timely manner, which is what I'm concerned about, right? I want these drugs, and I want them now. I have all this information and all the knowledge, but if we can't go from what did you have on your slide, helix to health, or it doesn't help any of the drug in it now. So there's some of those regulatory things we'll talk about later in the day. I think it's critical to move in this from the information we can get.
I think, from my point of view, trying to integrate all of that data so that we can actually do statistical analysis, machine learning methods on it is just critical. The New York Genome Center is working a little bit backwards on this. We're working with PCORI to integrate clinical data records from 6 of the largest medical institutions in New York, and I think there are 2 more on the waiting list right now. We're collecting all of the clinical records, the longitudinal medical records for all patients de-identified. So they aren't consented yet, but they're de-identified, and we'll have all those records, and we can query those records for various, earlier medical or environmental exposures. What we don't have yet is the model for how we're going to combine that with genomic data. We're certainly going to have to get patient permissions and re-consent patients to use that. But we're starting out saying we're going to need deep clinical data, and that's the hardest part we have right now. So let's get the deep clinical data, use that to find the patients and consent them, and then hope we can consent them to get genomic data and allow us to integrate the data.
So let me ask a question about the genomic data, because this, as somebody who is, as a journalist, read clinical trials results in the medical journals for many, many years, you realize there are p-values and tendencies in outcomes, as opposed to this works, this doesn't work. The lay public doesn't often realize that. Things that work for 60% of the population or 70% of the population. As I've read into some of the genomic research, you get this distinct impression that it's the same thing. We're identifying tendencies within these variations that are associated with disease states or maybe the natural history of somebody's life. And so how certain are we as we look at genomic information? As there are certain things, the low-hanging fruit have been identified. We're constantly searching for new variations that are associated with diseases or disease response or possible drug responses or whatever, but they fall within a statistical pattern. In other words, maybe 60% are like that, or 70%. So how are we, what is the state of play in understanding what it is we're looking at when we're looking at genomic information?
So I think, at least in pharmacogenomics, the way that that's often expressed is what percent of the variation that you see in a particular outcome is attributable to genomics versus environmental effects. And I would, of course that varies by the particular treatment. And one of our, the best models for determining how much, environmental and genomic data affects a particular dose is actually in warfarin, where we know we can get up to 40 to 50% in terms of explaining the variation. But that also, there's, a fair bit of uncertainty, there's, a fair bit of that variation that we don't understand. So some of that is unexplained genetic variation, and some of that is behavioral data that we just don't have. So it may be how the patient's taking the drug, or what are the foods that they're eating that may be interacting with the drug. So all of those things affect, a particular outcome. And again, it's this problem of integrating genomic data with other data types to get a complete picture. So there needs to be a lot more research to do that kind of work on other drugs if we're going to explain all of the variation in drug outcomes.
How about just in your center, trying to identify new variants that can be associated with specific diseases?
So I guess there's a couple of different categories here. The first thing I should say up front is we don't understand well yet the problems of penetrance and modifiers, where you have
Might want to explain it.
yeah, I know. Let's look at BRCA1, okay? It's a breast cancer gene. The first information that came out about it said if you had 2 copies of the BRCA1 mutation, or one of the mutations, there can be more than one, you had an 85% lifetime risk of getting breast cancer. And then we discovered that, that initial study, it was done on Ashkenazi Jews with a family history of breast cancer. So all of a sudden, if you take away the family history of breast cancer, we're not sure what the penetrance is. And there are people who think that if you're not Ashkenazi and you don't have a family history, then your risk may be in the 30% range, almost down to normal. But we don't really know because we haven't sequenced enough of those people, because we can't find them, right? Because if they don't have breast cancer, we don't know they have the gene, right? Nobody's gotten enough genome sequenced in the general population. There was recently a study that flipped that on its head, and they looked at Ashkenazi Jews with no history of breast cancer and discovered that if you had the BRCA1 gene, you still had an elevated risk of breast cancer, even if you didn't have a family history. We don't know what's contributing here. For those who don't know, Ashkenazi Jews are this category, a subpopulation that's studied in genetics all the time because all Ashkenazi Jews have very similar genomes. They're a very inbred population. There are some other very inbred populations, but it was sort of the first one that everybody did studies on, and all of a sudden it looked like Ashkenazi Jews had a higher risk of everything. They were the only ones studied. And so this notion of studying the genomes of people with disease makes it very hard to understand what the penetrance is in the population until you have genomes from people who aren't sick. So we don't understand modifiers, and we don't understand penetrance. But I also want to say that environmental factors aren't completely separate from genomic factors, because a lot of the environmental factors cause genomic changes. They can cause what we call epigenetic changes. Smoking causes genomic changes in your lung cells. So there isn't a split there. And we have to figure out how to put that together. It's an enormous big data question. It's an enormous computational problem.
Peter, it seems to me that that very same issue has been playing out in your community in cystic fibrosis. You just talked about your own 2 children have 2 very different effects. In thinking about that, is that an environmental response?
Obviously not, because they grew up in the same household, right? So it would be tough to say it was environmental. It's almost certainly more complex. It's systems, right? So our systems are complex, and we modify genes, whatever's going on in there. I think that's the ability of big data to help us get insight into those. Why is one person different from another? And I think, we talked a little bit about this applying other data, both clinical data in cystic fibrosis is a big issue around compliance of medicine, around health, direct correlation between weight and nutrition and lung function. So being able to pull in all this other information, correlate it accurately with the genome, I think is also where there's a huge amount of benefit, but that's going to require more data, bigger data, people understand how to link those things together, and more complex privacy issues and concerns around those as well.
So let me do a follow-up question for you. Because this community is one that has probably, I think the breast cancer community is another one that's worked a lot, and especially in the BRCA1 and BRCA2 gene, but in the cystic fibrosis community, this has been going on now for almost 2 decades, or a decade and a half rather, where there's actually been people collecting data and thinking about this. Just kind of walk us through the evolution of that, because it seems to me maybe it's instructive, because there is this sense I get sometimes that this era of personalized medicine is right around the corner. Once I've identified my gene, I can go out and act on it. Certainly, when you look at what Angelina Jolie did, for instance, it creates this sense that I should act now on some of this stuff. Whereas when you think of that from a traditional perspective, like clinical trials, trying to get information that has been actually validated in a scientific manner, certainly from a regulatory standpoint, there's going to be a lot of talk today about that. Has the community learned through this process to be, I don't know if the word patient is right, but to have a more sophisticated understanding of the issues that it confronts?
Yeah, I think, when 25 years ago, when the gene was discovered, everybody had a big party and said, great, we're done, right? We got it cured. And then all of a sudden, a couple of years went by and everybody realized that didn't do anything for you. Just because you know a gene, so what? If you look at what Vertex have done to get this drug Kalydeco and the other drugs they have coming out, they really applied technology, high-throughput screening. Understanding what the genome was and understanding what the protein was allowed them to use high-throughput screening to hammer away at it. Right? So when Kalydeco came out, they didn't even know how it worked. They just knew it worked, which is fantastic. And then what we are going to see, I think, the combination of genome information and high-throughput screening and more of the compound libraries is a significant shortening in the amount of time to actually get effective drugs available. From whatever it was, 20 years, 25 years to get a drug to market. Kalydeco was done, I think, in 12 years, something like that, from lab to FDA approval. And that amount of time is going to continue to shorten, which is great. Brings a whole bunch of other issues, but they're good issues to have, and everybody in this room could contribute to doing that.
Dr. Peterson, I'm going to make you the proxy for the practicing physician community here this morning. So, we do stories in our website and our magazine all the time about the difficulties physicians have with using electronic health records. People are hiring scribes out there. There is literally lobbying going on. I'm coming here from Chicago this morning. A steady drumbeat of lobbying by the AMA and others to postpone meaningful use, to not, put the actual requirements into effect that the government had hoped for when it poured out over $20 billion of electronic health records. Given that setting, what is the likelihood that physicians are going to find this actionable in ways that are meaningful to them? What will it take? Let me ask the question that way. What will it take to create an environment in which this will be actionable information for practicing physicians?
Yeah, very good question. So first of all, with respect to sort of implementation in EMRs and physicians, I think actually there's lots of things about EMRs that physicians really like. And you have a really hard time taking that EMR away. What I think they're objecting to a lot of times is prescribing how you use the EMR, what to do with it. That's part of the objections to meaningful use. So with respect to genetics, everyone has recognized that it's probably almost impossible to practice genomic medicine with these kind of high-density data sets without the use of EMRs. And so then the question is, what are the aspects of the EMR that are actually helpful to practicing physicians? So one of the things that we've discovered is that, first of all, making important genetic effects just visible in the EMR may be one of the more important things that you can do. Now that doesn't tell the practicing physician exactly how to use it yet, but it puts it in front of them so they get some understanding over time across multiple patients what that information means. And secondly, we've used a combination of classic clinical decision support, which might be a separate screen that they encounter when they're prescribing a drug,
along with pharmacists who are very well versed in the science to actually help them make their decisions. So I think that it's hard to teach physicians with just sort of electronic screen, but to have the combination of the screen and an educator that's available, that's been successful for us.
So the EMR vendors, Epic was mentioned this morning. They're one of the three major ones. And Geisinger is interesting as a system because they do have this closed network. And whatever area of healthcare you're looking at today, they're on the front edge. So let's look at the electronic health records there. Are the records themselves ready for this? Or do we need another bolt-on to our electronic medical record system to accommodate genomic information?
Well, we definitely need a lot of modifications to the current generation EHRs to include genomic information. Most EHRs are really not ready for it. You get single gene reports that are scanned into the record. Those are essentially not retrievable down the road. And if you're talking about a panel and it's all scanned in, most of that panel information is going to be very difficult to access. So the current generation is not adequate. The places that have Epic and Cerner in our own institution that have been translating some of these genomic results to clinical care, we've sort of made our own bolt-ons or created aspects of the EMR to sort of fit it in a reasonable fashion. So everyone's developed a little bit of a different solution. I think going forward, we're obviously going to have to have a more universal solution. Epic tends to have lots of different kinds of implementations at each center, and so at some point they're going to have to sort of bring sort of the best implementations and hopefully roll them out across all of their EMRs.
Don't let me put my editorialist hat on. I've written several times about interoperability and the problem with all the vendor space. Dr. Bloom, I'm very interested in what's going on in New York. I mean, you're dealing with 6-8 institutions. They probably all have different electronic medical records. So how you, in doing your research, what difficulties have you encountered and how have you overcome them?
In pulling together all of the electronic medical records, those electronic medical records don't have genomic data in them yet. And we are going through the process of coming up with standard dictionaries and standard templates for how to translate the data and translating them from each hospital medical system into standard formats and then loading them into a single database. And we do have data from 3 of the hospitals loaded right now. We still have 3 more to go. We should have that within the next 6 months. We should have those full medical records. The more interesting thing that we've encountered isn't actually moving the data. It's each hospital deciding what we can and can't do with the data, and what data we can and can't see. I have had literally 8 lawyers in a room arguing over what de-identification and re-identification mean in HIPAA regulations. Okay, and that, I mean, that has caused us more trouble. That is what's making it so hard for me to find a path to combine that with genomic data. That's why I sort of said we were at the nexus of all of the different stakeholders who need to be part of this. Everybody knows we need to be able to do this. We need to be able to protect the patients. Everybody wants to protect the patients, but how do we integrate the data? Right now, New York State actually requires not even electronic. They require a written informed consent signed by a patient to allow them to be contacted to participate in a genomic study. They can't even just be contacted. They have to agree to be contacted. And so, oh God, Geisinger sounds so good. And so we're struggling with that. I mean, I've put in requests to have all the hospitals contact patients with specific diseases just to sign a form that they have to sign in ink that says that we're allowed to have a physician call them to ask them if they want to be in a study.
Now, has this been driven by the lawyers, or is this what you think really HIPAA requires?
I don't think HIPAA requires it, I think New York State does require it.
Just in New York State?
Okay, I'm at least getting a head
It's not,
shaking here.
it's not a HIPAA requirement.
It's not a HIPAA requirement, I believe it's a New York State requirement, which supersedes HIPAA.
And so
Although I will add that HIPAA lawyers, and I confess I'm one, are among the most conservative people in the world, not including myself. But so many times, I can confidently say HIPAA doesn't ban something, and you'll find a lot of HIPAA lawyers who say, well, in our institution, we like to be more careful, and so they go way above and beyond HIPAA, and that is a very common disease afflicting my breed.
Can we talk later?
You bet.
So, through our mics, if other people have questions, we'll start taking them. But let me turn it over to Peter for just a second. So over time, was did that problem ever emerge in dealing with the cystic fibrosis community? Both questions that emerged. One is about allowing my data out, which is the second question. But the first question is just simply, they're all over the country. They're dealing with different electronic medical records. Where electronic medical records exist, I'm sure if you go back 10, 15 years ago, when you were dealing with this, there was nowhere near the diffusion of records. So how did you deal with those questions over time?
So you should see my filing cabinet. It's got x-rays in it, it's got floppy disks with something on it, I don't know.
And there's nothing to read the floppy disks anymore.
Not anymore. And so, yeah, I mean, we're talking about managing huge amounts of data with genomic information. We haven't even got regular electronic records sorted out yet. So good luck with that, everybody.
Thanks.
And the second question was...
Well, the question was, so how did you manage it over time? I mean, how did you create a learning system that got us to the point where we actually have a drug that treats this, given the kind of nature of this records that were out there?
So there's a huge discrepancy between the regulatory framework of HIPAA and what patients want. Especially if you've got a disease, you just want this fixed. You can have whatever you want from me and do whatever you want with it, just fix the disease. And I think we see this all the time when the doctors are like, we have to fill out all of these forms. And patient advocacy groups, in particular, I think have this balance between privacy but the need to share. You can go out onto any bulletin board, Facebook, whatever. You give me my genomic information, I'm putting it on Facebook for the whole world to see. And yet, in the industry, handcuffed to what you can and can't do, and we've got this discrepancy between regulatory and social right now. That is just where we are, I think. You see that in, a great example is with all the studies that are going on now, clinical trials. We've got a lot of clinical trials going on in cystic fibrosis, and you will see people put their information out on Facebook. Now, they're not supposed to, right? They're saying, hey, I just started this trial, and I feel fantastic on it. And which is a shock and horror from people who are in the business, right? But this is what patients are doing. They put that stuff all over social media, especially when the drugs are as highly effective as something like Kaleidos.
So, Dr. Bloom, you talked at the beginning of the session about having a place to share genotype and phenotype data. And as you well know, one obvious jumping off point might be the database of genotypes and phenotypes in NCBI, also known as dbGaP. So as we get more and more genomic data, what structural changes would you like to see at dbGaP to integrate more of this data? And also, as a user, how would you like to be able to use tools to process the data at dbGaP?
Very interesting question. So, two things. One is, and I will say, I was at the Broad for almost 12 years. During that time, it was my group that submitted about 40% of the data that's in dbGaP. So I'm well aware of dbGaP. You can go look at the numbers. Go talk to Mike Fiolo if you'd like to. Or I can give you other names of people who know as well. I may know your name, but not have met you. There are two things. The one thing right now is, it looks like dbGaP doesn't accept all data if it wasn't NIH funded. You have to get some sponsor. Which means that dbGaP can't be the only database we can use. The other thing I would say is that I can use dbGaP just fine. The New York Genome Center is this collaboration of 12 hospitals, and the number of doctors who've come to me, researchers from those hospitals, and said, I got permission from dbGaP, I can't figure out how to get to the data, can you download it for me? Which I will do as long as they put me down on their application so that I'm allowed to. But you know what else? I have this database in New York with all these hospitals, and I have, do you know how many researchers in New York have permission to use TCGA data? It's 2.5 petabytes, or it will be by the end of this year. I am perfectly happy to share that data with anybody who has permission to use dbGaP, but dbGaP won't let me do that. Although apparently if all of them put me down as a collaborator, I can anyway. The rules for being a trusted partner, both for NCI and for dbGaP, are too strict to make it easy to share data with people who can't afford to pay for terabytes or petabytes of data themselves. And I'd like to make it easier, and I'd like to allow those researchers to be in the same place so they can share their own data with each other, as well as with the big data sets, and that's not easy right now. And like I said, dbGaP is working, they've been enormously helpful in telling me how I can meet the regulations and do what I want. But it would be nice if it were easier and we could all share. And otherwise, other databases are going to pop up instead.
Thank you.
Thank you all very much. John Graham from the National Center for Policy Analysis. My question is driven by Mr. Guzner's concern or interest in this question of off-label prescribing, and Dr. Bloom, you said earlier maybe a drug that we know works for the variant, but there's no clinical trial. And Dr. Peterson, if I understood you correctly, said that's not really possible. There's so many variants that they're going to do a clinical trial for everything. So in the future, as this big data world emerges, do you foresee the regulation of clinical trials and drug approvals evolving such that some types of observational study could be promoted more towards the gold standard if we can't do the classical double-blind randomized controlled clinical trial? Thank you.
Yeah, so I mean, there, already we use post-marketing surveillance to change labels and to change the way we prescribe drugs. So, and that is oftentimes, not always, but oftentimes observational data. And usually it's related to safety concerns that you can't run a clinical trial on anyway. So there is sort of a model for using observational data to help make drug decisions. I think it's important to note that already a lot of the high-density genotyping is happening earlier in the drug approval process. So, I mean, the low-hanging fruit may already be defined by the time the drug gets to the market. It may be more the rare variant issue that we're sorting out after the drug gets to the market in the future. From my point of view, whether that rare variant is related to efficacy or safety, it should be able to be translated into clinical practice, and I think there'll be a process to do that.
I can give you a great example with Kalydeco. Kalydeco is approved for a very specific mutation of cystic fibrosis called G551D. We know from in vitro testing that it'll work on about another 1,000 people across 30, 40 different mutations. The clinical trials aren't being done for those because it's, there's just not enough people, not enough time. If you could just give them the drug and see if it works, that would be awesome. So if you can change that policy for us, yes, please.
Just to follow up on that, but you could give them the drug. It's an approved drug, and so the question, isn't that educating the physicians to prescribe off-label and to accurately collect the data so that we could create a learning system to see, whether or not the FDA ultimately will change the label, it will depend on the robustness of the data, I presume.
Yeah, so you can get a prescription off-label. These drugs aren't cheap. So the whole other segment of the puzzle here we haven't touched on at all, which is payers, right?
Yes, I did not ask you any questions about the Cystic Fibrosis Foundation and its decision to sell its property rights in the drug.
And it also may be that in these rare variants, there aren't enough patients to statistically prove effectiveness. Even if in a test tube you can show it and genomically you can show it, it's not going to meet FDA guidelines, and so what on earth do you do? There are people dying, but how do you fix that? That's why I said everybody has to work together.
So I totally agree with what you're saying, and I think that's where we need to.
Could you identify yourself, too?
Michael Levy with Pharma. I fully agree with what you're saying, and I think that's why we need to work with the FDA in order to determine where can observational data be used to make regulatory decisions, including changing the label and including getting approval for each of these rare variants so you can have coverage.
Yep.
There's a question.
I know I couldn't see over.
Hi, Tramplin from the National Institute of Allergy and Infectious Diseases. You talked a lot about the environment and how it impacts outcomes, and we're really focusing on the patient sequence data. But are there any efforts to look at the actual microbiome and how that could potentially affect outcomes or reaction to drugs and therapies?
We are currently doing an autoimmune disease study where we are looking at not only the genome, but the transcriptome over time. In fact, we're taking blood weekly to see how it affects, how RNA expression changes before and during flares, and we're taking microbiome data regularly to see if we can either find correlations in the microbiome in general or it changes as a disease flares and recedes in the remitting, relapsing kinds of models. So yes, we are looking at microbiomes in conjunction with lots of other data when we can.
I mean, sometimes I'm asked if we're using gene expression data along with the genetic data to translate that to clinical practice, and I think, you know, we are very excited about that prospect, but one of the things that is a struggle in clinical practice is just the complexity of the data. You know, our physicians, at least half of them tell us they really don't even want to see the gene name. They just want to see sort of the clinical effect, because otherwise it's just a bunch of letters and numbers for them. So I think introducing new omic classes of data is likely a very exciting field, but I personally don't feel like it's ready, you know, to start sending to the clinic.
One of the things we see in cystic fibrosis is actually looking at the genome of the pathogens, particularly mycobacteria, pseudomonas kind of infections, and really trying to dig down and understand a specific strain of them and where one might cause more issues with somebody with CF versus a different, you know, just a slight change in that pathogen. So there's certainly some value from extending genomics, not just in the patient, but the stuff that causes the problems as well.
Hi, it's Ann Waldo. I'm a health data lawyer with Waldo Law offices here in town. I'd love to follow up on the legal burdens to data sharing, because I'm very passionate about that. But my question actually comes from the other end of the spectrum from HIPAA. I'm an advisor to a Robert Wood Johnson Foundation initiative to take the vast amounts of consumer health data outside HIPAA, being generated by all of our wearables and lots of other areas, and somehow make better societal use of all of this data. And I think many people have the vision that in, you know, X decades from now, all of these amazing sources of data will all be integrated and put to great use. But my question for you is, what do you see that could be done in the short run to find value in the wearable data that's being generated? Like specifically, would it be useful to you and how?
So I'll answer that in two ways. There are researchers who are working specifically on that, and we're working with some of them, and I should give you those contacts. We are in this same study where I said we're using microbiome data and we're using RNA data. We actually tried to use Fitbit data. It didn't quite work, but we are at the moment using an app on people's cell phones to track how far and how fast they walk every day, because we think that, well, we don't think. We are testing whether whether their changes in movement would predict an onset of an RA flare before patients were really aware of it. there's some data out there that says you can predict a Crohn's disease flare from how people move. And so if we can correlate that data with changes, for example, in the transcriptome that we know change during flares, then we might be able to use that app to tell a patient that they're going into flare, they're in prodrome, they need to go to a doctor and get high dosage steroids now, not wait till it gets worse. Because I think there are often periods where they're not sure if they're gonna get sicker or not because they don't feel well all the time. If we can accurately use that simple data to tell them to go to a doctor, we can prevent permanent damage. We don't know if it'll work yet, but we're trying. And we're trying in conjunction with some folks at Cornell Tech who do this full time. And so I will try to get you in touch with them, if that helps.
Yeah, so I'll just add that we're not yet using wearable data, although we're anticipating that it's coming. But one of the things that I think about wearables right now is it's a pretty constrained set of measurements. And so in terms of the usefulness for research, we of course would like to see it expand. One of the areas that would be incredibly important for what I do is to get better measures of drug exposure. And so if there were apps or sensors or a combination of the two that would tell us whether someone actually took a particular medication and how much they actually took, that would be, leaps ahead of what we have in our current EMRs, which every 3 to 6 months or maybe every couple of years, that particular medication field gets updated. That's a very crude way of representing what the patient actually took.
I mean, one of the things I should say, Corey asked us if we could do was bring in streaming data from glucose meters and insulin pumps and combine that with the medical records so that they could monitor that. Haven't done it yet.
Okay, I want to, in order to keep this less on track than it might have otherwise been, I want to thank all of our panelists for, I think, what's been a very engaging hour. I think we did get at some of the promise of, and some of the problems as well of genomic medicine, so thank you all. And speaking for Joel, I'll take the moderator function here and say, you got 3 minutes to go get whatever you got to get, and then we'll get back here and get on to the next
section. Relics and Health and Life Sciences program, who's really working towards this goal of delivering personalized medicine at the touch of a button. So I'd like to join me in welcoming David.
Folks, good morning. I'm David from Intel. What I'll do today is talk about our vision of personalized medicine, and I think one of the first questions people have for Intel is, what is Intel doing in healthcare and life sciences? And I'm here to answer some of those questions, and hopefully give you a good background about genomics and links to a couple of books you should go read. Nice bedtime reading, but will give you a good sense about the policy issues in genomics and values in genomics. So, high school biology 101, for the folks who are not physicians or biologists in the room. We've talked about genomics today, but I just want to put this one foil up quickly. Words like DNA, RNA, proteins, cells, that's genomics. Right? That's the work that happens. And what you see here basically is what people in biology dogma, as they call it, that everything starts with the DNA, gets transposed to an RNA, and that becomes a protein. And it's a protein that sort of controls everything in your system. So you will hear things like proteomics, which is this next generation after genomics, and metabolomics, and I think they've heard a few words already. To make things easy, there's a great book you should read, and Valen's gives you a very good perspective about what genomics is all about. So this is a good, easy, I shouldn't say dummies for genomics sort of a read, but it should give you a good sense of what this thing is all about. Genomics, again, we've heard about it today. It's going to take a village to make this happen. The personalized medicine is going to be a combination of pharmaceutical companies working very closely, so the cystic fibrosis case, working with the NYGCs of the world, the Broad Institutes of the world, clinical hospital systems, companies like Intel, IBM, Oracle that are working on technology aspects of things, because this is a big data problem, to actually make this real and make this happen. So we've got to acknowledge some of the partners here. We've heard about this again today. When you go to your doctor's office, God forbid, with a positive biopsy result or some serious condition, data comes in multiple different batches to it. These are all silos, right? And the doctor can run a report, and you get this information within maybe 2 days, 3 days. I don't know if you have any cancer patients or survivors in the room, but someone who has just found that they have a positive biopsy result, 3 days, 4 days wait is not a long, it's a lifelong window. So what you have to do is, could we do all this in a day? Could we get that information out to the patient in a day with some package? And I'll give an example of what's happening in Germany, and that just set this tone of how important this is. So for us, personalized medicine and insight for us is this combination of multiple different data buckets. And it all comes down to technology to put these things together. And our intent is to make sure that the scientists and the researchers and the clinicians working in this space lead with science, lead with their research work, but they shouldn't spend too much time worrying about the computing aspects of things. And we'll show you in a minute, storage, how much it takes to store this information. And I'll give you a good start on that. You folks should be worrying about that stuff. That should just happen in the background. So that's personalized medicine for us. So it's going to be a combination from the bottom. This is your Fitbit data, your Nike Fuel Band information that's coming out, combining into medical records, to your genomics and pharma and clinical trial data, to provider information from hospital systems, medical records, for example. Some of the things we've been working on a medical record case is a lung cancer patient popped up in the hospital, and the doctor checked with him and said, how long were you smoking for? Well, the patient said, what do you mean? The doctor had pulled up the word smoking from that person's medical record, which was scanned for the last 25 years. This is in the UK, which gives a lot of data from the past. And the word smoking pops up on the record 15 years ago. Okay, so the doctor actually assumed that the patient was a smoker the past 15 years. The document actually said the patient stopped smoking 5 years ago. So he or she wasn't smoking for the past 20 years. So the physicians, as all of us know, have 10, 12, 15 minutes with the patient. I'm being generous here. It's much shorter than that these days. But these sort of information can send them down the wrong track. So how do you pull that out? So now, when genomics comes into the play, into the mix, and you saw one gene versus 3 million combinations of genes, it just makes the problem intractable. So that's personalized medicine for us. And the end state, all of us are in healthcare, we know, is the quality of care, the access of care, and the amount of care that's delivered for the patient. I think we have seen this. It's about 18% GDP in the US is spent on healthcare, and I think any health conference you have to show this number. It's one of those little moments you have to share. The second piece is aging. People are getting old, and all of us, some of us are older than a few of you here, but the hospital systems aren't ready to handle that. Right? We can't pop up a new $2 billion hospital system every other year in a large city. That's just not feasible. And so a lot of the telemedicine elements or remote patient monitoring become important. Now do that when you go throw in things like genomics and wearable data and all the payer information, things are going to get complicated. The third piece is, this is a McKinsey report which shows you the value of big data in health and life sciences. It's a staggering number. I mean, we know, wastage in healthcare is a problem. We have those numbers, but applying these sort of big data techniques to these solutions does help to sort of save. And the smoking case I told you is another example, because the physician would have said, "Hey, go get another scan to see what's the impact in the lungs," when that treatment wasn't needed. That's the case with VAS. This is just pure storage. This is the amount of information that's stored in hospitals today. Without the genomics information, these are all your images that are stored. Everybody knows what this 4K technology is all about. This is beyond high definition TV. So now in the hospital that we work with, they give out, once you do an ACL surgery, they give you a disc with 4K movie of the surgery. 4K technology is going to launch next year in the world. People are demanding that they want that high definition stuff. Bottom line is data. This is all getting stored, and I got an ACL done 12 years ago. They gave me a VHS tape for people who know what VHS tapes are. Things are changing, and this is just one hospital where the data is really growing. This is zettabytes. You will hear words like that now. If you heard the words gigabytes today, megabytes. That becomes, I believe, exabytes, petabytes, zettabytes, and yottabytes. Yoda from Star Wars. That's the future. Genomics, again, a lot of data. If you listen to sequence cancer patients in the US alone, it's a pretty big number. Newborn screening, again, pretty big number. And any genomics folks in the room, this third image that I put is a mandatory image that you have to put in every genomics event. This is the cost of genomic sequencing is down to $1,000 a pop. It was almost $1 billion 13 years ago. So just see how things have changed in that little window. And well, all that's done is the cost of sequencing is $1,000, but the money it takes to go process that information that David talked about, for example, is $10, $20, $30, $40,000. So that has to go down as well. I guess Toby is in the room. So is she here still? No? Oh, so that's fine. The Broad Institute, which is where Toby used to work for before, is one of the large genomic centers in the United States. They produce 200 petabytes of data. Don't worry about what the petabyte stands for, but just see in contrast to Amazon, Microsoft, and Google, how big that number is. If you thought Facebook was cool in terms of the amount of data it produced, think again. So that's the impact of this technology is going to bring to the data. And now when you combine this with your medical records, and think about a physician who is willing to spend 10 minutes trying to read through the data, that's the complication that's going to happen. So political issues, regulatory issues, barriers are all great. I mean, those could be solved. This is another problem that has to be solved in this space. David talked about 100,000 genomes getting sequenced. It's what, 200,000, 300,000 down the road. That's guys in the United States, yeah. But if you just look at the world, this is a worldwide phenomenon as we speak. You talk about the Ashkenazi Jews, for example, in the Middle East, they all emerged from 3 tribes. So the Middle East is now launching these huge sequencing programs, trying to sequence up to 100 million people. Right? If we thought 100,000 was bad, the 100 million does too. But the bottom line is, the data that's popping up in all these places are in silos. Right? United States, for example, has a pretty mixed population from around the world. I'm Indian, my data is compared against a standard US database. You may not find a variant for me, but you might find a variant for me because we have some ancestry back from the Middle East. You will actually see that I would rather look up that database. Can I do that from the United States? I guess I could because the data has to come here for processing. Well, data cannot leave geographical boundaries. Right? In Germany, for example, data cannot leave the country. What happens then? Well, the solution is kind of easy that I send the question to them and then they send me an answer back, rather than they shipping all these large amounts of data across back and forth. The thing at the bottom again, it's actually a map of all the sequencers. Sequencers are these devices that are used to sequence the genome. You can see the hotspots around the world. And that's actually a real-time map. So Illumina should be happy because a lot of sequencers are getting sold worldwide. But data like that make this a complex problem. David and the panel did address some of the barriers, and I'll just sort of quickly summarize what the barriers are. On the clinical side, looking at all the lab results, and I think you did mention about the lab data and accessible to the lab data. CLIA is the certification for the lab. In a nutshell, what that is when you get a blood drawn today for a cholesterol check, when it goes to a certain lab request or something like that, when the data comes back, a cholesterol of 200 actually means a cholesterol of 200. You can actually be sure that that is 200. So it's a control, it's a valid lab, and the data coming back from it is real. Now, genomics today is in the research phase. All the data that's generated is for research purposes. Research does not have to rely on a CLIA compliant lab. Research does not have to rely on a CLIA compliant lab. So what happens is now when clinical traces come into play, they have to go through a CLIA certified lab. And that means all the CLIA things that come with it. The second piece again is training. It's getting the masses educated on genomics. And literally to the extent that I'm a sort of an engineer by background, but I sit on tumor boards now. I've been doing this for about 4 years with cancer patients now. So it's the training thing that people can understand this process really easily. That's not a problem. But you need these training programs and classes, and that's set process of actually getting people trained. And as David mentioned, he had to tell his doctor what his report actually meant. But that has to change. On the technical side, it's all about the data getting created, getting generated. Data has to move from place to place. It has to be stored. And again, back to research versus clinical, as you all know, clinical images today have to be stored in 5, 7, 10, 20 years, right? Because for clinical purposes. Imagine storing this genomic data for 5, 10, 15, 20 years on top of the imaging data, right? Yes, you have to get sequenced once in your life, but we know environmental factors do affect genome sequences. So you might have to do it maybe every 5 years. If you go to another country, you spend 4 years there, you have to get it sequenced again. I don't know, but you have to keep the data together. Ethical, of course, there's a big deal with GINA. The GINA process took about 12, 13 years to build out and actually get rolled out. But there are provisions in play today, so that's not a problem. But GINA has its own problems. I think we've heard about that in terms of getting long-term life insurance and mortgage payments and things like that. There are some loopholes in GINA, but there are provisions, so you can build upon that. Clinical consent, who gets access to the data? Now, back to the cystic fibrosis case, you would put the data on Facebook, but your first cousin, who is also on your Facebook account, could get pissed because now you've just exposed potentially his or her genome on your machine. Like you just made it public without talking to them because you're part of the same family, isn't it? So those issues will start coming up as well. It's good to share data, but again, with whom, how long, can you monetize that information? All those aspects will come into play. And of course, the big US healthcare question is who's going to pay for that information, who's going to pay for healthcare. And I think as reimbursements actually change, that's the tipping point. One good sign is, and again, folks, correct me if I'm wrong, but CPT codes for healthcare do cover genomics now. So if you want to get a whole genome sequence, there is a CPT code, I think 1, 4 covered, 1 almost 5, 6, 7 digit numbers. But that's a step, that's a first step, that these things are coming into play and whole exomes all getting broken into pieces, but that's a first step, things are changing. Again, a quick recommendation on this book. I will tell you, the main essence of the story, but Henry Lax was the first person whose cervical cancer, I think, cell lines were used, and that is the reference people still use today. She's one of the first cell lines that people are using. But read the book and you'll find out the issues around ethics, issues around monetizing the information, how pharma companies have made billions of dollars on that single genomic line, but our family hasn't seen a single dollar. Does that, so again, it's good to share for research, but again, if people want to monetize your data for research, shouldn't you be paid for that? And so naturally that comes with its own provisions and accesses. But again, it's a very interesting book to read about the fundamentals of what the barriers in this space. So now to some solutions and what Intel is doing in this space. This is this health system in Germany. It's called the Charité Health System in Berlin. What this actually was the first project we did, I think, 3 years ago. So we sat down with the German health system and said, in your hospital system, medical records, lab results, radiology data, tumor banks, specimens, biobanks, blood banks to work. So we put everything in this one big system, one big database. And literally what took a few days happens in seconds. And every physician, all of us in the room, we know you need an iPhone app for everything these days, right? You need an iPhone app, an iPad app for everything. So there is an app for this. The intent of having an application for this is it's an iPad app, and Angela Merkel, who's the Chancellor of Germany, actually demoed this at CeBIT. CeBIT is one of these large consumer electronic shows in Hamburg. And the intent is simple. The physician at the bedside of the patient can pull this data. Right? He or she doesn't have to put up the laptops and log in 20 times and try to get answers for the patients. There is an app for it. Right? That simplifies things a lot. So that's one of the positive things in this space. The second one, again, the details aren't important, but creating these devices, these standalone devices, just to do a certain operation, right, is always a good thing. So it's like a microwaves at home, right? It's been designed to heat up your food. I mean, that's its main job. I was in Korea at Samsung a few months ago. They have this new microwave coming out that actually you put any food inside it, it figures out how much time it should be around for. You put a slice of frozen pizza in, it takes images, it processes it on the microwave itself, it says, frozen pizza, defrost more in 5 minutes, done, stuff. Why can't we build devices like that? I mean, that's a really bad example, but devices like that to sort of simplify these things and get it so simple that a physician, I don't want to say a patient is a slice of pizza, but you stick it in something and it just figures it out, right? All comes out of a bit of data saying, and IBM Watson is one of the directors in that step. You feed it enough information, it's learned from, thousands of pizzas, made worldwide, and it has all the data, thin crust, everything, and it pops up an answer to you. Or it gives you a few options, saying based on what I've seen, option A has an 80% chance of success, so try that, or option B or C. The answer is not to give the physician an answer, but a range of options based on what it's seen. So again, this is with Dell, with TGen, it's a pediatric oncology system in Phoenix, and that's the case for this one. Cloud computing. I'm sure all of you have heard the word cloud computing and Amazon Web Services and Google and Microsoft Azure. This is a case where if you need additional compute for a certain application, but just need that for a few days, right? You don't have to go buy 50,000 machines just for that one little burst of compute. So just go to the cloud. Now, cloud has its own issues. You don't know where it's gone, right? Patient data, would you want to send it to the cloud? Amazon has done processes in place where they do business associate agreements with the hospital system, so there is a trace and audit trail and all of the good stuff. But as a physician or as a patient, would you want to send the data to the highly open cloud? In Europe, for example, again, if it's Germany and you want to use the cloud system, Amazon doesn't have servers in Germany. It's in Ireland. So the German government can't use Amazon in Germany. So, right, the data is just left above it. So those are the issues that come into play. But I mean, cloud is a great example to burst out compute when you need it, but it has its own options. This is a case in China, and the reason why I wanted to put it up here is we talk about interoperability of health data, and that being a huge problem in the United States. The smallest health network in China is 3 million people. Okay, electronic records actually flow from Caribbean up north all the way down to Shenzhen for immigrant work, if he or she wants to practice the data. The main reason putting this file up here is that problem is doable. You can do that, but the Chinese government started from a clean sheet of paper. In the United States, we have so many legacy systems, be it the NHS in the UK, in Canada. You have to bolt on things now to existing infrastructures. That's the problem. Right? If we could just sort of start on a clean sheet of paper, this problem is easy. You can put 300 million records within 6 months, you're good to go. But that's the challenge we have to deal with. But again, you can fix existing systems or bolt on stuff to it to make this real. This is possible. This is not difficult. So in the UK with the NHS, for example, the National Health System in the UK, you can add these additional clinical tools to go process existing data that's already in those systems. That's possible, that's doable. And you apply simple techniques like Hadoop. If you've heard the word Hadoop, big data, using this yellow elephant, and that's the simple things. It's all about the brand these days. What Hadoop does for you is searching through large amounts of data. Right, so let me give you an example. If I had a deck of cards, 50 cards, and someone told me, find me ace of spades. Right? There are 2 ways I can do this in this setting. One, I can start searching, which will take a while, or I can give each one of you one card and say, who has the ace of spades? Well, the answer is right there. Right? So how do you search? So there are techniques like this that are coming into play that another case is when you try to look on a laptop for a certain file, and if the laptop happens to have, I don't know, 500 gigabytes worth of video images or something, right? It's going to take you a while to find that file. But then again, if you had lots and lots and lots of small files and have to search through those files, there's a whole different way of doing things. So again, think about how you can apply these different compute techniques. And again, these are techniques that have been used in financial services. Right? I mean, Goldman Sachs makes millions on these nanosecond trades. Why can't we use that for cancer genomics? Right? These things exist out there. So let's find, at Intel, that's what we try to do, is get these different domain expertise. I think Oracle has a whole big database as well. How do you learn from these different verticals and put these things together? Back to training. So what we've done at Intel is we conduct training programs. This is with the CDC and Georgia Tech. This is to train folks, I think, in infectious diseases. This is post the Ebola scare. I call it a scare. It's not an outbreak, it's a scare in the United States at least. So the intent was to train PhDs in biology, PhDs in diseases, MD and PhDs, in how to use compute technology. So you can have a combination of a PhD, and again, on the flip side, you have people with PhDs in physics, PhDs in computer sciences, who want to help do these things. Because at the end of the day, it's just a data, it's just ones and zeros. It's in some game. So these classes that we conduct, 4 days, 5 days, actually, hopefully the intent is, after the 4 days, the computer scientists and the bioinformaticists and the oncologists hopefully speak the same language, right? Or are able to ask at least the right questions, so we don't get sidetracked. So we do this very often at the Oregon Health and Science University, where some of us from Intel actually are visiting faculty there. We have oncologists today writing software programs because they know the subject, right? They know what needs to be found. And people like me who are on the other side of the fence can now sit in tumor boards and sit with oncologists and say, I think that cell is mutating based on whatever I've seen in the last 3 or 4 years, or the books that I've read. So this is the next wave. We need all of us to sort of bind and do this together. And finally, the policy papers that we have published, and again, these are Intel point of view papers. And just a quick shout out for the Twitter folks who are following data-driven med. We have actually posted the link to one of these papers, so take a look, because that does address a lot of the barriers from our perspective, especially clinical, ethical, less technology, because we hope you understand that, but do check that out. And on the same lines, we do have something with the European Union as well, and that is going through the EU system, so when that gets ready to publish that. At the end of the day, one of my previous slides, this is a problem that everybody's going to see. Right? Somebody has to step up and take the lead in this space, and could we call this a moonshot program? Right? What Kennedy did in the 60s to send someone on the moon. Could we be the first ones to sequence and create this infrastructure, get everything sequenced? A final quick tidbit: wheat, rice, is different seeds that we have. They have better sequencing techniques than humans today. Right? I mean, all of us know, right? The wheat seeds can grow in the middle of nowhere. How on earth did that happen? Let's not use the 3-letter word called GMO, because that's a bad word, but there are cases where the thing actually works, right? But the funny part is the whole wheat genome hasn't been sequenced yet. That's a different story. But the intent is, if the agribusiness can get this right, we can produce the world's best seeds and rice grains. I mean, especially in China, the amount of rice research that goes on is mind-boggling. They can grow rice in Antarctica if they have to. So, why can't we go for this cancer? That's, I'm not trying to trivialize the concepts, but the techniques that are used there, here. In Sweden, you have people studying spruce trees and how, they spend billions of dollars trying to understand these trees and how they grow. Maybe there's a usage model for that, don't get me wrong, but there are techniques that you learn from that, because at the end of the day, the gene is a gene is a gene, right? I mean, it's the same concept. And the interesting part is, David mentioned, the humans have 3 billion base pairs. Wheat has 7 billion base pairs. A spruce tree has 21 billion base pairs. So if you can get the problem right for 21 billion, why can't you get it right for 3? Is it complex? Yeah, it is, but techniques do exist. So let's collaborate, let's share, and drive these sort of policy changes to, open up, silos so people can talk with each other and sort of make this happen. So with that, I'll stop. Personalized medicine division. The time I get to a hospital with a positive result to, my physician telling me that, yeah, go try those 2 clinical trial drugs, and I think you have an 80% shot of, something would happen to you. You might feel better. Takes months to do, right, with all the cocktail drugs. The current, line of sight is 2050. You will have all that done in a day. But at least I can't wait till 2050, and I think some of us in the room can't wait till 2050. So, could we get them by 2020? This is going to be this big mix of the village, as I talked about. That everybody has to play this game Of course, you're going to have business motives and things like that, don't get me wrong, but there's got to be that some level of contribution that we have to do to the human race and human society. Let's play fair, let's play nice. ClinGen, GAP-DB, these are all these NIH-funded initiatives. Let's play in that, and let's contribute. And that's what David is doing, and contributing all that stuff into back into the system. Let's do that. All right, with that, I'll stop. Thank you very much. Questions? Yeah, simple enough.
It's a nice talk. So it seems like we have enough computing power available to interpret large volumes of genomic data, but what about sort of the software side? I mean, you're working with hospitals, do you find that that's where most of your time and effort goes to, or as opposed to sort of putting in the computing power essentially?
That's a very good question, and the best example is so Wisconsin-Madison, co-badgers, where I did my grad school work. I wrote a piece of software in 1993 for genomics. They still use that software today. I'm dating myself now, it's 20 years ago, but people still use that. So that is a problem because that thing was written for this one old Apple machine which was built in the late 80s, early 90s. Today, these machines which we have, if you go back in time, you needed maybe a million computers to do a certain job. That thing gets done on a laptop today in a heartbeat. So stuff has to move over time. So all these hospital systems don't want to change something that's already, which is not broken. Or a lot of these hospitals are spending all this money on getting these EHRs installed now. So efforts like this are getting pushed out, but software is the biggest challenge for us. There's no question.
I guess I want to add a comment on the workforce side, because I see workforce as being a real serious challenge. And at least in my experience on interdisciplinary work, it took more than a few days to get the language and get people calibrated across disciplines. And in fact, some instances, I think you have to have people dually training in dual competencies in bioinformatics and biostatistics, for example, and so forth. I guess my concern is that I think as we got into the high-tech, we've essentially built a lot of 6-month wonders, if you will, who call themselves informaticians, and you do need those people. I don't mean to denigrate that, but we didn't invest in the upper end, high end of this, and I think we have a huge need in clinical informatics and research informaticians. And I'm just interested if you'd flesh that out a little more on the workforce side, how you see it.
I think the answer to that question is, training people to do this. And I'll give an example at Intel, for example. Most of the folks we have who are working on genomic programs have improved genomic software almost 100,000 times, right, in terms of speed and things like that. Came from the DreamWorks animation team at Intel. They used to work on all our accounts managing animation for stuff down in LA. These are the guys who actually built How to Train a Dragon. I'm sure all of the kids that watched that movie. These are the guys who actually did that work. But at the end of the day, it's a data problem. So you have to start getting people out of these different specialties, getting them trained for the right, like clinical informaticians, for example. Folks who have been doing payer analytics, people who work at your Cignas and Latinas, who have been studying premiums and things like that for the last 25, 30 years, are perfect informaticians. They just have to be trained in HL7 codes. Well, some of them do know what the HL7 and CKD codes are. So it's that you're going to train them and get this, but we have seen in the last 4 years, really anybody can be trained. I mean, I'm going to trivialize on the oncology side, but we can speak the language. It takes time. Doctors don't trust us, but you can give them 2 more data points, saying, hey, you thought these 4 things, let me add 3 more to that. I think workforce training, I think, is, but it's not a difficult problem to solve. My opinion. Okay. Thank you.
Thank
Our next speaker is going to be Dr. Jane Yu. Dr. Yu is a senior advisor for healthcare and life sciences with an IBM Global Business Services and Worldwide Technical Computing. Dr. Yu has more than 20 years of experience in clinical healthcare, biomedical research, and advanced analytics. And she holds MD and PhD degrees from Johns Hopkins University. So please join me in welcoming Dr. Yu.
So hi everyone, thank you so much for, I feel I'm absolutely honored to speak about a topic which is incredibly exciting to me, having been a clinician and computational biologist in the past. I'm going to start off with a story. Some of you might have heard this story because it was published in the New York Times in 2012. It's a story about a patient who in 2003, at age 25, was diagnosed with acute lymphoblastic leukemia, or ALL. Now, the patient was diagnosed at age 25. He received a standard course of chemotherapy, successfully went into remission, and stayed in remission for about 5 years until 2008. He started noticing symptoms. He relapsed, and survival from a relapse of AL is not very good. But he underwent another course of standard chemotherapy, had a stem cell transplant, and that was successful. He had some complications with respect to that transplant, but all in all, he was in pretty good health until about 2011, his second relapse. This time they tried a standard course of therapy. It didn't work. And it really didn't look like this patient was going to make it. But so, in the bottom corner of my slide, you see a picture of a medical scientist and medical oncologist at the Washington University of St. Louis. His name is Dr. Lucas Wortman. And what's interesting about this doctor, is that he's also the patient that we've just described. And in his second relapse of ALL, what his colleagues at the Genomics Institute of the Washington University of St. Louis decided to do was sequence the genomes and study the proteins that were being expressed in both his tumor and in his normal cells. And what they found was interesting, that the gene FLT3 was found to be completely out of control in his tumor cells. What was also interesting is that FLT3, there was actually a drug approved by the FDA called Sutin that targeted FLT3. And what his colleagues and friends at the Genome Institute decided to do was give their colleague Sutin off-label. And what happened was pretty miraculous. When they thought he was on pretty much, was not going to make it, he recovered and he went into remission, and he's still in remission to this day. So I want to read you a quote from Lucas Wortman. I can't overstate the importance of those discoveries that were really driven out of the research lab, but made their way just in a matter of weeks from the research lab to helping me as a patient. With new technologies we have today, we're now able to decipher the way small changes that are present in my tumor genome, which may actually look acutely different from someone else's. Changes in my tumor genome that were unique, that led to changes in my treatment. In 2003, the same year that Lucas Wortman was diagnosed with ALL, was also the year that we completed the Human Genome Project for approximately $3 billion. It was a project that was conducted over 13 years. But as was mentioned in the previous talks, the cost of whole genome sequencing has gone down from $3 billion for one human genome to only about $1,000, and it can be done in a single day with new technologies. So what we're here to discuss as we've heard all morning, is how understanding variations in our genomes, that unique sequence, A's, G's, C's, and P's, how changes in or variations in those sequences have impacts on our clinical outcomes. And the idea here is if we understand those relationships, then we can develop drugs that target drugs like Sutin that can target or have an effect on biological processes that involve the different variations in our genome. Between 2005 and 2010, the change in personalized medicine investment rose 75%. Also, during this time, bioinformatics companies were investing in personalized medicine research. About 94% of them were. And between 2006 and 2014, the number of prominent personalized medicine treatments and diagnostics that were available rose from 13 to 113. Now, a large part of the personalized medicine research is funded by NIH programs like the National Human Genome Research Institute, they're funding 3 large-scale genome sequencing and analysis centers, one at the Genome Institute at the Washington University of St. Louis, where Dr. Workman was fortunate enough to receive his treatment. The other 2 at the Broad Institute of MIT and Harvard, and then also the Human Genome Sequencing Center at Baylor College of Medicine. What you see in this chart are also, each one of those little blue dots represents a medical research institution that received funding from the Clinical and Translational Science Award that's also funded by the NIH to institutions that are showing promise and excellence in bringing laboratory findings to the bedside. There are about 60 institutions who are receiving funding through that. But as our previous speaker noted, I mean, this is not only a U.S. issue. This is actually a worldwide issue. And in fact, it's an international healthcare priority. Just this summer, the UK invested 300 million British pounds in Genomics England, a 100,000 genome project, which is their nationwide push to encourage investment in life science and pharmaceuticals within the country. David Cameron, their Prime Minister, during a launch made this statement. This agreement will see the UK lead the world in genetic research within years. I'm determined to do all I can to support the health and scientific sector to unlock the power of DNA, turning an important scientific breakthrough into something that will help deliver better tests, better drugs, and above all, better care for patients. The UK, through their 100,000 genome project, plans to sequence these patients, 100,000 patients with cancers and rare diseases by the end of 2017, and they plan to integrate that data into their National Health Service electronic medical records. Also, the little nation of Qatar in the Gulf region. About a year ago today, Her Highness Sheikh Moza bin Nasser, who is the chairman of the Qatar Foundation and the wife of the former Emir, launched the Qatar Genome Project. What she stated is this: we're creating an environment conducive to research and education and linking research to clinical practice. I see personalized medicine as a revolutionary way to diagnose and treat patients, treat diseases in the future, and this is the vision of Qatar. The country of Qatar's national vision is to transform itself from an economy based primarily on hydrocarbon fuels into one based on knowledge. And what you see here is a picture of a hospital called Sidra Medical and Research Center, which in 2014 just opened its doors. Brand new hospital, has a high throughput genome sequencing center as part of it. And their goal is to sequence the genomes of very large populations of Qataris, if not the entire Qatari national population of 250,000, trying to understand how they can develop better drugs and treatments for illnesses that really plague the Gulf region uniquely. But perhaps the biggest player on the international stage is in China. Beijing Genomics Institute, founded in 1992 as a participant in the Human Genome Project. Now, back in September of this year, Francis Collins, the director of the NIH and former director of the Human Genome Project, made this statement. The United States and the Human Genome Project, and from that derived a great benefit. The return on that investment was 178 to 1 in terms of economic growth. Yet if you ask me now, where is the largest investment in genomics in the world? And it's not the United States, it's in China, Shenzhen, China, and they read our playbook, and they saw that the opportunity was there, and they took advantage of it. BGI currently, it became an official Chinese state agency in 2008, and currently has more sequencing capacity than the entire US, and 1/3 of the total global capacity. In addition, as you can see in the, on the map, they also have centers in Cambridge, Massachusetts, and in Copenhagen. And just in 2013, they purchased a Mountain View, California company called Complete Genomics to do for their genomic sequencing analytics platform. But regardless of where this work is happening in the world, I and my team at IBM have had a real privilege to talk to the leadership, not only at BGI, at Sidra Medical and Research Hospital in Qatar, Genomics England, and many other biomedical research institutions interested in conducting personalized medicine, not only within the US and all over the world. And one thing that's been very clear to us is that this market, despite all of our progress, is still very, very young. And the reason it's very young is that they're all struggling with the same problems, right? And those problems, you know, highlighted to some extent from our previous speaker, so I'm going to have to thank him very much for making my job a little bit easier. There are 3 main areas where we really see difficulty. The first, from a technical point of view, is big data. You've been hearing it all day. We need to combine genomic information with clinical records. Not only, it's clinical records taken over the course of a patient's history, no matter where they've been treated, right? And we need to combine those. And there's also another source of information that we can leverage, and that's work that's been done by previous scientists. That's current facts that are hidden in peer-reviewed journal articles, textbooks, you know, unstructured or medical literature, textual information. We can actually leverage that information not only to validate our scientific studies, but also to find the potential inputs to a computational problem. So just to give you a sense of scale, we need data on thousands of these patients, genomic and clinical, in order to come up with a statistically valid research project. And just the omics alone, the raw data, 200 gigabytes, just to give you a sense of scale, the computer that I'm running this presentation off of has 4 gigabytes. So to hold the data for a single person's genome would require 50 of these computers. Right? The second issue that we're dealing with is data silos. We have medical institutions capturing this data, but they're scattered all over the world in systems that are very different from each other and that don't talk to each other. And not only are we facing the problem of having to integrate data across different geographic locations, but we're also having to integrate data within the 4 walls of a single institution. So each research lab within that institution might have a preference for a different research technical platform. And so we need to figure out how to get that data to talk to each other as well. And the third area that a lot of scientists have had issues with is the fact that most of the data that we're capturing is unstructured. And by unstructured, I mean that it can't be captured easily in an Excel spreadsheet with numbers or fixed text strings. Unstructured is something like clinical notes. or medical images, large swaths of text, peer-reviewed journals, websites, social media. So there again, we're trying to figure out how are we going to extract information from unstructured data. So what makes this even more complicated is a challenging business environment in which we need to conduct this data. And this is not only in the academic world, but also in the commercial world. And I would say particularly among commercial pharmaceutical companies who we are speaking to a lot. And one of their problems is the threat of extended R&D timelines. Now, as we spoke about this morning during the panel discussion, the low-hanging fruit is gone, essentially. The easy drug targets are gone. It's no longer one genomic variation, one disease. We're now talking about multiple variations that can possibly affect disease. So because we have a much more difficult scientific problem, it's going to extend the timeline of your research and development efforts. The other thing is that these diseases are much more rare. So what's happening is that you need to find patients for your clinical trials. So how do you find patients if hardly anyone has them? How do you find the thousands of genomes and clinical records that you need to do a viable study? So there's a lot of concern about recruiting patients to these clinical trials and creating a valid study. Further delaying the R&D timeline. Time is money. And the last issue is concern about more stringent regulatory requirements that are delaying the trials approval process. Now, at the same time, you're thinking, okay, I'm spending X amount of money for my R&D operations. How am I going to get that money back? Okay, so there's a lot of revenue uncertainty. And there's a Battelle study out stating that worldwide they project about a 3% increase in the life science R&D spending and funding. But is that funding adequate to actually, is that funding adequate to recoup the money that you've already spent on that extended R&D timeline? And the last problem is related to greater competition. So there's this fear about data exclusivity and IP and shortening those timelines. So as a pharmaceutical company, I'd be interested in making sure that I had my IP rights, secure long enough to recoup the extra money that I spent on those in R&D. So there needs to be a time, a IP in place long enough for me to recoup that money. Okay. So what is IBM doing in this space? Well, one of the areas, one of the possible solutions, we're working on both compute and storage infrastructures to support personalized medicine and translational science worldwide. But one of our, our highest profile technologies is Watson. And you're probably familiar with Watson. You might have seen Watson beat Ken Jennings and Brian Rutter on Jeopardy. Well, what exactly is that? Well, to explain that, what I want to do is just go through an example. One of the types of unstructured information that we have to analyze is clinical data. And I'm going to look at this patient history. It's in the brown box, and I'll read it to you. The patient was in good health until 2 or 3 months ago when he gradually developed fatigue and intermittent epigastric pain, and recent colonoscopy that was performed was within normal limits. There's really important information in just that text. First, gradually developed fatigue. The fact that the fatigue was gradual and not acute is diagnostically significant. The fact that it was intermittent and not constant, that's also significant. All right, so that's going to have an impact on your diagnosis. All right. So, and our previous speaker paper gave the example of smoking. Smoking, all right, the patient is a smoker, the patient smokes versus the patient quit smoking 15 years ago. There's that word smoking. It's not a simple text mining task, all right? You need to understand where smoking sits in the context of everything else in that document, or where fatigue or epigastric pain sit in the context of the rest of that document to really understand it. So that's what Watson does. What Watson does is based on set semantic rules, it scans unstructured information for patterns in the language. All right, and so what you see in this, right beneath the Watson machine in the slide, is an example of a clinical record that's been annotated by Watson. Watson goes through that record and sees that, okay, each one of those highlighted areas represents a clinical concept, like smoking or epigastric pain. All right, and then it transforms that into a structured code that you can, so a structured format like this, correlation matrix or heat map on the right, and it uses that matrix, essentially numbers, to do a calculation that can be used for research. So you take unstructured clinical record, turn it into a structured form that a scientist can actually do a study on. That's what Watson does. And this form of Watson is currently being used at MD Anderson. Here's another form of Watson, another application of it. Now, as a medical student, one of the things that you're tasked to do is memorize tons and tons of information about the diseases that might afflict your patients. And as a doctor, as a responsible doctor, one of the things that you're expected to do is keep up to date on all the medical literature. What was underlined in this text on the right side of the page is keeping up with the medical literature can take as many as 160 hours a week. So I'll tell you, as a, having been on the wards and doing call, at times 48 hours at a time, the thing that's probably top of my list over that 48 hours is going home to sleep. And I would say that most of my colleagues would agree with that. Right? So we don't have 160 hours to comb through all the new medical literature, and that's what we're putting Watson against. We're basically using Watson with its semantic rules to scan millions and millions of pages of unstructured literature. And what it can do, it can do that annotation job. It can find patterns in the language, millions of volumes in a matter of seconds. And that's how Watson was able to win at Jeopardy. That's how we're applying it to the healthcare space. So one of the things that I want to make a note of is that Watson does not profess to be a physician, or it's not going to produce a diagnosis for you. It's not going to tell you what the right treatment is. It does not take the place of a real doctor. It's a support, it provides support for a real doctor, but it doesn't take the place of a real doctor. What it can do, however, is point you to diseases or therapies that, as someone who just didn't happen to read the article, that you might have otherwise missed. It tells you that Sutin is a potential treatment for ALL. Okay, so keys to accelerating scientific breakthrough. We've been hearing about them all day. From a policy point of view, what do we need? First of all, in order to do this type of research successfully, first of all, standardization of clinical and life science data. Second, the ability to develop technology solutions that are capable of processing what are rapidly growing volumes of complex life science data. Third, scientific collaboration and data sharing in a secure environment. Incredibly important given the fact that there is no longer any low-hanging fruit. And the third is intellectual property protection and financial incentives, because the cost of research and development in pharmaceuticals is huge, and it's getting larger and larger as low-hanging fruit disappears. And so what we need to do is make sure that R&D organizations are incentivized financially to continue innovation. And so with that, I'll end my talk. If there are questions.
just wanted to ask, if you're doing one of these analyses of unstructured data, can you incorporate a foreign language? Yeah, because if everything's going on in China, you'd want to crisscross.
We do, we do. And we do have labs within IBM that are working on foreign languages. We have a group in our Ireland lab in the Dublin area that's working on foreign languages. We have, we apply there are a whole host of languages. I don't know if Chinese is one of them. That's something that I would have to look for specifically, but yes. Actually the Watson development team is in Tokyo, Japan. So not natively English.
Quick question about intellectual property.
Yeah.
How concerned is IBM about the Patent Office's approach and the court's approach to appear to be limiting biotech patents?
Well, to tell you the truth, it's not something that I'm familiar with. So that it's something that I could check on.
So one more question. So some of the techniques you described will come with some uncertainty. So for example, like language processing or defining new genomic or phenomic association. So how do you represent that clinically or to the researcher? Is there a confidence interval or what's the sort of approach that you take?
So, there has to be some sort of confidence interval associated with it. That is a good question. It's really, what we do is really to provide the infrastructure to do that type of analysis. How the researcher wants to use that information, how they want to grade the certainty of it, what is the threshold for, trusting this piece of data, that's something that the researcher is going to have to do within their own computational model.
I'm sorry, I got in here a little bit late. You may have already covered this. Anytime you get involved with medicine, there's always a lawyer somewhere involved. Does Watson carry are you looking for Watson carrying malpractice insurance?
No. And that's a question we get asked a lot. Watson is there as support. It's not. We don't assume it's not a Dr. Watson. We don't assume that Watson is a doctor, right? The decision, the final medical decision, has to be the physician. And this gets back to the uncertainty issue, right? You don't know. There's a probability that what Watson brings up is correct, but we don't know if that's correct. And the physician is going to have the final say in that scenario.
That doesn't stop the lawyer. It's out of pocket.
Right. These are issues that that are very real, and that we do need to deal with. Because that issue does come up quite a bit.
There's really fascinating
Senior Vice President, focused on molecular diagnostics, genomics, and personalized medicine. He's also CIO for HITECH, a for-profit subsidiary of the Moffitt Cancer Center. He has over 30 years of experience in the pharmaceutical, biotech, molecular diagnostics, medical device, and CRO industry. He's had a number of senior level positions in a variety of fields. He has advanced degrees in cellular and molecular biology and in leadership and business administration.
Including the people who are doing the language
Good afternoon, guys. I'm not sure which it works. Having to talk to keeping you for lunch, during lunch, right after lunch, or just before you go home. So I got all three to make it work. And in the meantime, when the senator shows up, we'll probably have to stop anyway, because I'm going to talk for the rest of the afternoon. Because what I have to say is the most important thing you're ever going to hear. So realistically, we'll just have to be flexible as time progresses through the course of the day, and hopefully have some time to use the Q&A and go through some of these pieces. But I do want to thank all the previous speakers that made my job a lot easier. There's a lot of the things that were raised, questions and topics that were brought up, are a lot of things that we're wrestling with within the Oracle environment, but also things that I had to deal with from a user's perspective through the different companies that I've engaged with throughout my career. I'm not an IT guy, so before anybody asks, I'm not an IT guy. I'm a user. In the drug industry, a user is probably a good thing, not a bad thing. But I'm in the CRO, biotech space, and the CRO space for more than 30 years, and predominantly looking at finding easier ways to make all that easier. Predominantly some lazy scientist, so I look for trying to find things to make everything go faster, easier, better, and hopefully save some money as we work forward. Being a publicly traded company, safe harbor statement, I'm not a sales guy, so anything I say cannot and will not be used against me. So what we'd like to do something wrong? Oh, okay. You never know that when the IT guy shows up and starts fiddling with something, and if you guys actually see something or hear me or whatever. All right, so you guys heard a lot about this morning about the scope and depth of the problem from a lot of different perspectives. In seeing what really is happening from the perspective of actual care delivery, and as well as from the perspective of therapeutic development, and what sort of transpires and key issues and things going on with just some basic things of how you share data, where do you put data, how do you normalize data, what happens to the data, who owns the data, which is one of the biggest things that we run into on a number of different occasions. So the complexity is actually quite big. And what we've seen in some of the different pieces that are going on is that our lives are becoming much more difficult and they're not getting easier. And as we move down the pathway through the course of time, as technology advances, it's going to get worse. The question then becomes how fast do we deal with the changes in technology and platforms. So as we've seen, we talked about the cost of sequencing going down. It's about $1,000 now, more than likely in a few years it'll probably be $500. Either that you can go down to the local CVS pharmacy and try to get them yourself to have a machine behind the counter. It may be a little bit too simplistic, but there'll probably be a lot of things that are occurring during that same time frame. And the other key thing is this real-world data bits. Big data versus real-world data, maybe it's an interchangeable term, but at the end of the day, there's so much out there. The real question is what's valuable in the scheme of what's, the wheat from the chaff kind of thing, to try to really understand. And then being able to aggregate those data to understand it. And then, as we're seeing it, me on the pharma side, grew up in the pharma space and predominantly in global clinical development. And if you think back to what we were doing over the past 25 years, more or less, we haven't changed clinical development that much. It's been the same sort of processes and scenarios we've done for the last 25 years. It's only been recently that we've gotten a little bit more creative in the schemes of where we are, and predominantly that was a lot to do with the economic environment that we lived in 2007, 8 through 11. So we had to do something different, and we had to look at things differently from the perspective of the whole healthcare policy environment. So in looking at how that works, it's changed the dynamics of it. Likewise, when you're looking at patient stratification and opportunities, again, historically, we just took any drug, put it into a population with the indication intended to be treated, and hopefully we had enough numbers to demonstrate that there was statistically significant differences between the control group and the experimental group. And at that point, hopefully it was enough to be able to get regulatory approval in the space. And then at that time also, the payers were not really jumping up and down and screaming at you for comparative effectiveness, but that as long as it was approved, safe, and effective, you got reimbursed for so much. Obviously, that whole process and activity has changed dramatically over time, and effectively dealing with the technology that's going on. So this slide is actually kind of busy, but it's the core context that I really wanted to bring into this is that center circle. In the perspective of that is that no matter where you are, no matter what you do, no matter what side of the fence you're sitting on, the only thing you have to really worry about and deal with is the data. Everything else is perfect. But the other key thing to take away from this is that everybody uses the same data. You know, we think it's different, we think it's unique, we think it's ours, but in reality, it's all the same data. So if you look at on the left-hand side of the slide, predominantly that's where the healthcare delivery system's been sitting, really looking at it in the perspective of what is going on with the care of the patient. And also looking at it in the perspective of this silly little thing I'm sure you guys are worried about, I'll call it accountable care, and this whole perspective of how you dictate, and also from the perspective you look at the institutions themselves, how do you make sure we're not losing money? Historically, it was all sort of moved down the same pathway, but really looking at it in the perspective of saying, how do I do a better job of actually effectively treating the patients and making sure that my institution is running effectively, efficiently, and productively, and I'm not losing my shirt. So we look at it in the perspective of what's going on the left side, it really is trying to understand more about the healthcare analytics, and outcomes at a clinical setting. We look at the right-hand side, that predominantly has been in the pharma sector. Pharma, biotech, CRO space, really trying to understand what you're really doing with the drugs, with the data that are being generated, and how you get to an endpoint where you can get an approvable entity that actually has a benefit to a patient sitting in the doctor's office. And we talked a little bit more about the bottom right-hand box about the interoperability of data and where it goes and how do you deal with it, and being able to share data. Because again, we've all seen the perspective saying that it's great if I look at my population of 10, 20, or 100 patients, but really what I need are 10,000 or 20,000 or 100,000 patients to really understand the dynamics of this population, to really make a difference and understand what's going on. So at the end of the day, is that you're looking at this in the same context, it's all the same data, but you're looking at it from different users and different perspectives for different purposes. And then the big data bits, the real world data structure and what's happening to the lack of structure of those data, and where is it coming from, the wearables and all kinds of bits that are coming into play now, better personalized, essentially personalized managed care. What are you doing in your perspective of what's going on? And one of the things that I always want to bring up into the scenario, into the volume, velocity, variety, and the value kinds of scenarios, is when you really think about it, it does get back right to the individual patient. I just had a personal experience with my mom, and I'm sure you all have one, who fell, unfortunately, but okay, was fine. But I found out after she went to the hospital that she did not have a primary care doc, contrary to what she told me. And what I also found out is that she was paying more attention to what her friends told her at lunch of what to do with her therapeutics than what her physicians told her what to do. Has that ever happened to you? Have you ever noticed that? So when you think about the real world data, what's going on at the patient level, and try to understand the environmental factors associated with that, think about the social environmental factors of their daily life to try to find out what's going on. When a friend says, well, if you're feeling good today, just don't take that because if it hurts your stomach. Or I got a better idea. I found this great pill in the local pharmacy, in the health food section, and it does amazing things. So when you start thinking about all the different dynamics that occur in this whole schema, when you start thinking about the big data scenarios of social information, what are you really getting? In the perspective of what's really happening in that space. So, and this has been sort of the perspective of what we normally would see in a pharma development regulatory approach environment for getting a new therapeutic through the process of approval. A fairly linear process, going through the middle where you have a bunch of molecular biologists and pharmacologists sitting in a corner someplace, some lab in the basement. Hopefully they don't come out because generally their social skills don't sort of match up to the normal environment. And hopefully something very, very nice pops out that moves through the development, you actually do something and get it sort of approved, which then you sort of go into the post-marketing surveillance business and try to follow and find out what's happening to that drug in a given environment. And as I was saying before, that whole process is no longer sustainable. Because when you think about when I was doing development for oncology drugs, specifically for solid tumors, we would take a drug, go into phase 2, phase 2B, and we would set up a program in solid tumors to try to hit as many solid tumors as we think could get into enrollment trial, and hopefully one of them or two of them would demonstrate some sort of beneficial effect. That whole process just doesn't work anymore. It's not sustainable, it costs too much, and the outcomes are simply not there. So we look at it, what the result of that was, is we have physicians have an armamentarium of drugs in their quiver that for the most part don't always work. Right? And they work in some of the time in some of the patients. And then the level of effect varies by patient, even if it is quote unquote effective. So when you look at these are just some of the examples, you know, of the different percentages of patients that actually respond to a given entity. And in the oncology space, it's generally around 30%, give or take. But if you think about the implications of that, what you end up doing is 60–70% of the time giving somebody a poison that doesn't lead to any good. You know, so it's a whole different environment in this case. And then when you also look at it in the perspective, we did a lot of work at the Moffitt Cancer Center and then with a number of different medical oncology groups around the country, where we really found out about how patients start coming back to the doctor. And what we found, especially in breast cancer patients, was they tend to be a very educated group of people. And often cases, they will come to the medical oncologist and say, this is what you're going to do. This is the program, I've read it, I've done WebMD, I've done all this research, this is what we're going to do. And a doctor sits there and says, well, I've run the genetic test, and I found that, you know, you don't need chemotherapy, you don't do this, or whatever, or you do, or whatever. And then they say, well, no, we're going to do this. And if the patient or the doctor doesn't want to do that, they say, thank you very much, and they go down the street and find another doctor who will do it, of what's happening. So the doctor shopping event really occurs. And then you also look at it saying that the genetic tests that are done aren't black and white. They give you some level of understanding of what's going on one way or another. The genomic health test, great test, gives you high, low, but about 35% of the time gives you an intermediate number. What do you do with the intermediate number? Well, some of the patients and physicians say, well, I'll do something, lower dose, shorter course of chemotherapy, doesn't do it good, but they all feel better because something's happened. So, you know, at the end of the day, some of that stuff is just to the point where how costly does it become to do some of those things when in fact we're not hitting the right patient with the right drugs. So this gives us, again, just a little example of the kinds of things that are going on across therapeutic areas. So it's just not unique, but again, going back to what we used to do to develop drugs, we put in enough patients into a clinical program to get statistically significant numbers that demonstrate an effective control versus experimental group, and, you know, wave the flag and call victory and get things move on and get the drug approved and on its merry way, when in reality it doesn't always work. So what we're looking at in the perspective of at least how we're viewing this within the Oracle environment, and some of the technology and approaches that we've been wrestling with, and some of the things that I've lived through the course of my career dealing with, is in the perspective that the drug development process is no longer linear. It truly is a continuous feedback loop mechanism. And when I was in graduate school, we had a professor that took this course, and particularly in organizational development and design, where you're really thinking about how organizations work. And he was saying that no matter what implementation that you do, you have to continually do a review process. We call it take a dare. You do assessment, diagnosis, action plan, implement, and review, right? So historically, what we had was a medicinal chemist in a basement in a lab facility doing something nice. What we're finding is that the actual feedback loop mechanism that actually comes from the data at bedside is finding its way back to the medicinal chemist. And the things that we're finding from the clinical laboratory at the hospital are finding way back to the medicinal chemist and the pharmacologist to give them different perspectives as what the targets should be, how they should be, and what the differences and problems that clinicians are facing at bedside, driving back to what's happening at the early stage discovery. So looking at it in the perspective, it really has changed the dynamic. And when you think about the economic environment we all live in, the idea of finding better drugs that have a better outcome for a given patient at a better cost, and not giving somebody something that doesn't work, changes the dynamics on an economic standpoint, not only for the patient, but at the hospital level. And some of the key things are looking at, again, the big data from the perspective, sorry, not big data, but just overall analytical data for processing hospitals. When you think about it, you know, especially the docs here, the hospital administrators say, you gotta get a patient out in 3 days. Goal, get them out in 3 days, if not sooner. Right, but what happens if you find out that if the patients and the given indication, we did this work with a couple of large academic institutions, and if you look at given indications, if you push people out in 3 days, well, we found that about 82% of the time they came back within 30 days. If you kept them in 4 days, that percentage dropped about 22%. So then the idea was, well, why would we push them out in 3 days when economically it makes no sense and they come back within 30? You know, so there's accountable care is having a major impact on what's going on. Likewise, if you think about how you care for a patient through a given algorithm of drug treatments, drug A, then drug B, then drug C, then drug D, because they're, you know, they're dictated either by formulary or by payers or whatever. Well, what happens if 90% of the time that the patients given drug A return and have to get transferred to drug B? So why did you give them drug A? This doesn't make any sense. Likewise, when you're looking at it from data analytics and thinking about usefulness in hospital settings. And what do you do with that? And how do you effectively wrestle with that? And there's some data that we generated, some time ago, looking at OR theaters and the costs associated with OR theaters and the processes associated with that. So some very interesting information derived from that kind of stuff, and how do you effectively use these data. And I'll be happy to talk about it if we have some time. Some of the stuff we've already heard, and the things going on over the past 5-7 years, effective use of genomics and targeting patients. These are some drugs, obviously, that have benefited from that, and people are benefiting from it today. These are some data that were published by a small pharma company down the street you probably know called AstraZeneca, which is very, very enlightening data. They took a retrospective view of about 10 years of data from their own R&D laboratories and looked at what was actually happening in their laboratory. And what they found is that in the development programs that are going on now, when there was an active biomarker part of the phase 2 program, they were successful 82% of the time versus 30% of the time previously. Likewise, when you look at all the different programs that are going on, 85% of the time had some sort of personalized healthcare strategy engaged into them, which is just a 400% increase over time. When you think about that, the impact of that is quite dramatic. Now, this is fraught with pluses and minuses. From a pharma marketing sales perspective, I don't really want to know this, because what I want to find is I want to get as many patients as I possibly can with the drug that I can, so I make as much money as I can. When you think about this in the perspective of the target, therapeutic approach, we have to start looking at it in perspective of incidence and prevalence. So we start thinking, especially with like the cystic fibrosis of the various disease states and the various sort of mutations that are approaching that. If I find there's 100 patients in the world, and it costs $1 billion to do this, what do I do, right? How do we start wrestling with this? So these are some of the critical issues that I know from my experience in dealing with the FDA, that they're very conscious about, very cognizant about, and they're very worried about, in the perspective of trying to look at developing these drugs to treatment of, in some cases, very small patient populations, even below orphan populations in some of these cases. So we look at it from the perspective of personalized medicine. We also have to keep in mind of using the data is there's a difference between personalized medicine and precise medicine. You want the precision from the perspective of populations, so get a really good idea what needs to get done, and then look at the person sitting in front of you. And how do I utilize all those data from cohorts of populations to effectively treat the patient sitting in front of you? So this whole construct of being able to effectively do this is that truly you're looking at the right patient with the right tissue, the right safety, right patients, and the overall, hopefully, the right commercial potential as you run forward. But that last item often becomes a bit nebulous as you really start to think about investment and opportunities and costs. So when you look at this whole bit, the significant challenges are still there, right? We still need to acquire the data, acquire it properly, and it's always, it's never nice and neat and like a cute little package. It's all from different disparate sources to do different things. So, all right, how many told I have to shut up now? Okay. See, I told you this was going to happen, and it's okay. We're prepared. I can stop here because this is actually a really good spot. Can I finish this slide? Yeah. Is it okay if I finish this slide? Real quick. Real quick? Okay. Just in the perspective of the across the silos and managing the different kinds of data. Because if you're thinking about like the omics data, we saw some of those things going up, but they're petabytes of data. They're huge amounts of data. We were shipping hard drives all over the country trying to use this stuff. Being able to effectively deal with those data and understand it is important. And then the interoperability issue. And also, I think the other key thing is the collaboration. Because the populations and cohorts are the drivers that we need to effectively address. So if that's one of the critical things, and I'll stop there so that we can then catch up next time. We'll come back.
Thank you. We will come back because that was a very interesting presentation. I do want to welcome to the stage here Senator Michael Bennett, who's a Democrat from Colorado. He was elected in 2010. He is the chief author of the HITECH Act that he introduced just recently with Senator Hatch, a bipartisan effort to promote innovation in health IT products by defining what's inside the FDA world, what's outside the FDA world. We think it would do a great job in promoting innovation. He's a member of the Health Committee, the Finance Committee, I would say that he is a pragmatic problem solver who hasn't stopped going since he's got to come to DC. So we're really happy, Senator Bennett.
Thanks. Well, thank you. I'm sorry to interrupt your presentation, particularly because you seem to actually know what you're talking about. These people are going to have to endorse somebody who doesn't really know what he's talking about. I promise I'll be brief. We are, somehow this place manages to slouch to the conclusion at the end of the year, at the same time that it speeds to the conclusion at the end of the year. They're colliding with each other, coming and going, trying to keep the lights on in what has become a land of flickering lights where, unfortunately, the test of for whether we're succeeding or not is whether we keep the lights on. And it's not, I think, a sufficient test of what we ought to be doing because the future is rushing past us and our relevance is going to be determined by whether or not we come to understand that. Washington is locked down in the past by incumbent special interests that are still fighting for the same benefits they acquired in the tax code 50 years ago or even 100 years ago in the regulatory code. And that is worrisome to me because it means that we're not focused on the innovators in our economy. That's why it's so important that you guys are here and what you're talking about today. I want to thank Health IT Now and the Center for Data Innovation for bringing you together, but I want to thank all of you for helping us think about how we're going to invent our future, because that's why we're here. Not here necessarily in Washington, D.C., but that's our job as Americans. And that's our job in whatever our chosen field is, and that's what a lot of people in Colorado are trying to do, which is why we have one of the most robust economies in the country right now, I think. And a number of years ago, when the bioscience community approached me from my state, and they said, Michael, we can no longer raise venture capital in the United States because it's all going to Europe and Asia because of the regulatory uncertainties of the FDA, and they said, can you help us do something about that? Richard Burr and I went to work on a new piece of legislation called Breakthrough Therapies, not because I had had a story from a cancer patient, although I've since had many stories from cancer patients, not because I had a story from a cystic fibrosis patient, although I've had many stories since then, but because I was trying to support a sector of our economy that actually was driving wage growth in an economy where median family income has been falling for the last 20 years, is making it harder and harder for people in the middle class and who want to get into the middle class to get ahead in this country. So the very elemental question for me was, how do we do some things, or can we do some things to make sure we can grow this, in our state, pretty nascent bioscience sector? And it turned out there were some things we could do, and it's now our third fastest growing sector in this state. 600 different companies that pay an average salary of $84,000. How do we have more of that, not less of that? You know, the large firms that are the ones that still have the benefits here are not the ones that are hiring lots of people. They're not the ones that are paying great salaries because they have become so productive and so efficient in the 21st century because of our use of technology, our response to India and China, because of the recession itself. It's the companies that are started next week and the week after that and the week after that. They're the ones that are going to drive job growth and wage growth. That's obviously a generalization, but you want to make sure that we're on the right side of that equation. So when you have a tax code that hasn't been updated since I was in college, the chances that it reflects the 21st century world that we live in is pretty remote. Just as it's becoming more remote every single day that the FDA is able to keep up with changes that are happening in our economy, and that's why Warren Hatch and I have been working on the HITECH Act. And we just think that our innovators need clarity that they don't have now. The FDA doesn't want to regulate things like calorie counters and Fitbits. Let's put it into the law and say so that you can have certainty about what your risk is that you're going to be caught in the regulatory law of the FDA, or the important knowledge that you do need to be regulated by the FDA because of health and safety issues. And if we can also better figure out a way for doctors to better analyze data, patients can have better information, we ought to figure out better rules of the road there and not just revert to a sense that somehow we're never going to figure out the privacy concerns here. Somehow we're never going to get the benefit of the incredible data that's out there this morning. I was with a company just today and saw the extraordinary data that they're collecting in order to have them make better judgments about their business, but also have their customers make better judgments about how they take care of themselves. And this conversation is going to need FDA, Congress, companies, patients. And I look forward to having this conversation in the coming Congress. The House has strong interest in passing this legislation, I think, and I think we're going to be able to improve it over here in the Senate. We have to be, as policymakers, I think, we're humble about the policy levers that we have to pull right now in this 21st century because of the velocity the world is moving. And as somebody who somehow ended up in government, I'm not quite sure how that happened. But we also need to pay attention to the fact that the speed with which the world is moving and the speed with which the private sector is moving creates a lack of synchronicity between the regulator and what's happening out in the world. And the metadata that's now available and analyzable with computer power that we couldn't even have imagined even 10 years ago raises very profound questions about how we regulate and how we think about what the role of government is going to look like in the 21st century. That's probably a longer speech than the ones you guys signed up for. So let me close by saying how much my office looks forward to working with all of you in the coming year to try to get this legislation right. And if you've got other brilliant ideas that nobody seems interested in, I hope you'll let me know because we're always looking to steal something from somebody. Is there time for a question or two or a criticism? I was an urban school superintendent for almost 5 years, so you can't hurt my feelings. A long time ago.
Senator, all this sounds very, very good, but are you aware of the efforts in intellectual property where the Supreme Court and the Patent Office are attacking essentially software patents and biotech patents? They're going, and is there a plan to do something about that, I hope?
We don't have a plan today, but I think people are going to look at it in the new Congress to see how it can be addressed and how we can balance the need to protect IP on the one hand and not do it in a way that degrades the chance for other people to innovate as well. And that's what we've run into is that challenge. I will tell you, this is unrelated, but on this point that I was making earlier about how we think about the regulator and all that, we were the ones that wrote the legislation that created the idea of having regional patent offices, because I didn't see why we needed one more patent examiner in Washington, D.C. Instead, have them out where the innovation is, and the second one of those is now in Denver, Colorado.
Thank you, sir. John Graham, National Center for Policy Analysis. Medical device excise tax repeal. When? How?
Well, oh my gosh. I think the votes are there in the new Congress. It's a question of, and what I can't predict is whether or not, what I cannot predict is what the pay for is going to be that's going to allow us to get this done. That's what I cannot predict. I know, I've been told that the new majorities, this is one of their first two priorities coming out of the gate. So I don't know the answer, but I have the telephone number of people that might know the answer. So I'll give it to them. Okay. All right, I got to go. I apologize. Thank you very much for what you're doing. We really
Thank you, Senator Bennett. Before you go, someone give him another hand to the med tech. We are going to not miss a beat and go right back to normal. Thank you. Now, listen, you guys get my building now. Well, if we could just do half of what he said, just half, or even 10%, I think we'd all become so happy we'd just go out and drink beer all day. You know, it would just be ideal. Hopefully it does happen.
But anyway, going back to this, what I wanted to be talking about is saying that the technology that exists today is, I can't say it's adequate to do all the things we need to do, but it is incredibly robust stuff that exists out there if we're given the opportunity to apply it and use it. And some of the restrictions, controls, and concerns that we all deal with every day, of data sharing, HIPAA, personalized health information protection, on and on and on, are all surmountable if we allow them to be surmounted. Well, what we've found in a number of things that we've been doing in applying these kinds of platform technologies across institutions, it's not that the technology won't do what we need it to do, it clearly will. But what we're finding is that the biggest issues tend to be the organizations themselves, policies, procedures, bureaucracies, and controls, in often cases, things that were built 10, 20, 30 years ago that almost are impeding the progress and growth for the next century, maybe the next 5 years. So what I want to wrestle with is in this particular slide, this is sort of like a 50,000-foot view of the world. To orchestrate saying that if you look at the left, the source systems, I mean, there's an incredible number of data source systems out there from all different perspectives, creating data in multiple different formats and structures that at the end of the day, we all want to use in some way, shape, or form. And again, historically, we seemed to be pretty happy working in our own little silo because it was our nice little sandbox, and we knew everything that was in our sandbox, and we could deal with everything in our sandbox. But what we're seeing in the perspective of essentially the larger picture of the integrated activities from the bedside to the bench, our sandbox and borders have just been blown out. So they're across the board. So the data that we're used to working with and very comfortable with is not only a fraction of the kinds of data that we actually do have to deal with and work with if we want to do our jobs and do it differently. So if you look at it in the perspective of whether it's a therapeutic development opportunity or a healthcare analytics process from a hospital environment of what you need to do to ensure that we're not losing our shirt, it is in the perspective trying to say, how do I deal with these data, deal with them effectively? So all the data sources on the left, and there could be a billion other data sources on the left to get to from any perspective you can imagine, and you put them into the bigger sort of stack of blocks in the middle. And if you think about it, and on the far side of the security measures of data integrity, robustness, sort of the one source of truth kind of a scenario to be able to get to, so that when analytical processes and tools people use to go out and look at data, if I do one query today and I do the same query tomorrow, will I get the same number? Will I get the same outcome measure? So the truth is sort of behind that is critical. So getting into analytical processes are way on top of that. But to get everything into the perspective of sort of the healthcare data model and omics data model, and in this case, the omics is not just genomics, this is any kind of omics. So metabolomics, proteomics, anything you can think of. And even genomics we got a better handle on, but when you really start thinking about proteomics and metabolomics, the numbers and the volume of data grows even greater in that perspective to try to understand it. The other key thing in that whole scenario, there are not a lot of standards associated with those data formats and structures, so it makes it even a little bit more difficult. But if you look at that base, those three boxes at the bottom, it seems pretty simple. All we have to do is put stuff in those three boxes and we're all done. The problem is that the heavy lifting to get the data normalized and standardized into those boxes on the bottom is a tremendous effort. And anybody here is working in any kind of large institution which has multiple data warehouses and or is trying to get data out of different warehouses we try to put together can fully appreciate how difficult that challenge is. It's not for the faint of heart in a number of different levels. So then on top of that is the perspective of all the analytics that you use, whether kind of healthcare analytics, bioanalytics, analytical tools that you can actually, that people use to effectively use the data to create information. Again, it's the structured and non-structured data that we heard about, the Watson activity. I mean, that's not trivial, guys. That's some pretty cool stuff to do that, to get the activities of being able to effectively deal with unstructured data. That's a huge endeavor to be able to really understand and effectively use that. And there's lots of ways to go at it, and a number of different approaches to go at it. But it's in the perspective of thinking things a little bit differently. We've talked a lot about EMR systems, and I hope you understand that the EMR system was built at a patient level, right? It's essentially trying to mimic a traditional paper chart. So it's looking at a patient level, and it's looking at data and information about a patient. When you start trying to look across patients, you start running into significant problems because the EMR systems are not generally built that way. And the other key thing is that the vast amount of let's say, the empirical or discrete data in an EMR system is very, very small. The vast majority of data are in notes. The dictated notes you try to quell up and try to pull from, which is an incredibly difficult task when you start thinking about EMR data to effectively use in a clinical environment. So in any event, this is again a very high level. This puts it a little bit more granular, if you will, where if you look at things like the bottom tier, and I did this on purpose because there was going to be a test on this slide afterwards, and because I'm going to ask you what the words are in the third box from the bottom. So the idea here is that this is a very complex piece of work that goes across multiple organizations' capabilities. Even he can't see, he's sitting right there. To be able to do this. Again, this is not a trivial task, as we all know. So trying to get this into some level of being able to effectively use, to look at the top row, where you actually start translating this into something that a physician can use, a hospital administrator can use, a researcher can use. In a lot of cases, they really don't care about all the stuff on the bottom. What we care about is the stuff on the bottom to give the user something they can use at the top. Right? So all the heavy lifting of being able to effectively accumulate data, store data, normalized data, standardized data is all done on the bottom. The users are, for the most part, they really don't care. What they want to do is show me the data that I can effectively use, R on it, SAS on it, and kind of analytical tools to be able to make some sense out of these data from disparate and multiple sources. So be able to and then portray it in a way that makes it easy for them. And we all know, doctors are incredible people in what they do. And the pressures on them to do the right thing and do it well are phenomenal. But then on top of that, they have to do it in 38 seconds. You don't have 160 hours to review medical literature, right? You just don't. I got to see 30 patients a day. I got to feel out find better ways to get patients engaged in their care, to be able to preempt a visit, and then follow up with them after the visit, so I can do a better job of managing that patient care in the longer term. Right? And there's some technologies involved in a way to be able to effectively do that and get patients engaged. But again, it's another topic for another day. So another key thing is really looking at the perspective of individual trial. And I'll go through this relatively quickly, but again, the idea is that if you're a healthcare researcher in academic institutions, you're looking at populations of the patients in front of you. If you want to go back to cohorts to be able to effectively look at historically in a longitudinal fashion to try to find what's common, what's different among those patients versus what's in front of me. In a clinical trial, you get a little bit better of a luxury because you restrict the patient populations. But you still want to start looking at what's in those patients to be able to effectively manage and control. And then, so again, this is also used whether it's a pharma study, sponsored study, or investigator-initiated study within an institution. You really start wanting to try to correlate the idea of the clinical data with genomic data to be able to effectively start understanding what's going on. And when you look at it after the fact, if you try to look at some cohort analysis and looking at populations and commonalities across populations, you really want to start looking at what's common across studies. And it makes it even more difficult to wrestle because you don't often have the same nomenclature or the same structure to be able to get to. So when you start looking at those, it gets a little bit more difficult to start wrestling with. But again, you need numbers to be able to start looking at what's common and what's different. And this is one of my favorite slides. And I spent a lot doing it because I built this. I really love it. Hey, I got pride in ownership. All right. So in this scenario, it's what I try to display is sort of a thought process that a clinical development researcher and/or a medical researcher would be doing as they look at data. Okay. So if you look at it in the perspective of anyway, those two yellow boxes in the middle, right? So if you look at it in the perspective of a clinical trial, you've got a control group and you've got an experimental group. And hopefully you have the numbers enough that they actually separate statistically and you demonstrate a clinical effect or a statistical effect of your experimental group versus your control group. But when you really look at that experimental group, it's not black and white. You got people who responded extremely well, and you got folks in that same group who didn't squat. They didn't do anything. So what's unique and different about the responders and the non-responders in your experimental treatment group? What's unique about it? And then you got all this dumbness of grayness in the middle with grades of effect over time. So the idea is that using genomics and other kind of other omics, you start creating some sort of demarcation. Maybe they're artificial cohorts, when you start looking at some artificial segregations of those groups to try to understand what's unique about them and what's different about them. Not necessarily say now have a biomarker, other than I have a suggestion of some signature that may in fact turn into a way to select patients as they're running forward. Now, in the perspective of this, I mean, this is research, right? So research by definition means I don't know the answer. So it's all probabilities. We're trying to understand how do I narrow my range of uncertainty. Little anecdote, I went fishing in Hawaii one time, and I always wanted to go deep sea fishing for marlin. I never did it before, wanted to go do it. There was a guy about 80 years old in the concierge desk. I wanted to go fishing, and he said, great. I said, what's the probability of actually catching someone? He took off his glasses, I sit down, and said, you know, if I knew that, I'd call it catching. We don't, we call it fishing. So when we're looking at this, we're fishing, right? We're fishing to try to narrow ranges of uncertainty. So when you look at this in the perspective of those cohorts, and you're learning what's going on, so you're narrowing your range of uncertainty. You do the next studies by manipulating inclusion and exclusion criteria, patient population selection criteria, and then you learn a little bit more, and then you learn a bit more. So you get to the end of the day that hopefully, when you get to phase 3, you actually do have a biomarker that you can validate with a sort of approach in phase 3 to get to an outcome and get to the end of the day and everybody's happy. Yeah, I won't go over that because it's already done. So the last piece here is just in summary again, the old way we did stuff is no longer sustainable. We've got to do a different job, we've got to do it differently, and the environment around us is forcing us down that path. We have no choice. I mean, all of us are looking at it from a different perspective, but we have no choice in what we do, whether it be patient care or therapeutic development. And the other key thing is that the therapeutic development process is no longer linear. It truly is an iterative loop. And if we ever stop getting the iterative loop, we're going to get stuck back into the thing that we were doing 25 years ago. It's simply no longer going to work, or another Me Too drug is simply not going to make it. The volume of data is overwhelming. The technology development is moving faster and faster. And we just heard from the senator, you know, we've got stuff in place from 30 years ago, regulations and environment, and we've got to deal with that because what we do every day, what everybody here does every day, is far and away surpassing what our regulators and capabilities can keep up with. The other thing, the second last bullet there is that we all know the process is changing, our environment is changing, policies are changing, and the one little bit I put at the bottom there essentially, you know, no outcome equals no income. So it doesn't matter whether you're treating a patient or you're the therapeutic area manager or director for a pharma company. The end of the day is there's no outcome, there's no income. You know, so we have to be thinking about patient outcome measures and having that cost effective in a way that we can deliver care appropriately and we can get to the right outcome, whether it be an improved patient outcome measure or we actually make money with a new therapeutic. So, you know, I guess, you know, the bottom line is it's pretty easy to get, you know, discouraged. You know, but again, we're in the infancy in this whole process of what we're trying to do, and the opportunity for us to really have an impact on this is just absolutely tremendous. So let's go forward and go do it. Thank you. We probably have 4 nanoseconds left for a question.
Did you catch marlin?
No, I didn't. But the boat next to me did. You know? I should have been here yesterday, you know? You know, I always heard that before. Yeah, caught 1,400 pounder yesterday. Yeah, sure you did. All right, sorry. Thanks again, guys.
Great, so we're going to move into our second panel, so I'd like to invite our panelists up for panel 2. So this panel is really going to dive into some of these data-related issues that we've already started talking about this morning, and really talk about some of the challenges around data sharing, interoperability, and analytics, what we need to do to advance that. So we have a fantastic panel here. Since I can't see the names, it's too well from here. You can see in the front, there we go. So immediately to my left, we have Don Denmark, who's a professor of medical education at the University of Virginia and a visiting professor at the Center for Health Informatics and Multi-Professional Education. To my right, continuing down is Paul Migueli, who's CEO of Pervasive Health. And then we have Peyton, who's been introduced before, from Intel, and then Mark Perlin, Global Vice President at Oracle, and RN, Dr. John Sung, Professor of Bioengineering and Physics at the University of Illinois. So I want to start off just opening up to the panel. We've heard a lot already this morning about the important opportunities here. We've started building up the IT infrastructure to now actually take advantage of this data. So I'd just like to get comments from everyone around what you think the current state of affairs is around data sharing, especially as it relates to genomic data. So Don, why don't we start with you?
First, thanks for the invitation. Yeah, I think I'm reminded of the comment that somebody, by now, everybody has said that almost everything once, but not by me. But to add to your question, I guess what I think we have is a growing sense of what we want out of all. But I think we're in the earlier stage of how to get there. And that's what I think we're hearing a lot today. So in a sense, what we're wanting is, I hear, is a health data ecosystem that will allow us to have a learning healthcare system for both individuals and populations, for all the relevant stakeholders. Now that's kind of wrapped it up, and that's kind of, I think, what we're saying we want. But obviously, how do we move to that is a socio-technical issue of great complexity. And it's the kind of thing in complex adaptive systems that you can't write the prescription. You live the prescription. And so I think there are certain kinds of things, boundary conditions that we need to keep in mind, and definitely some policy reforms that I think we have to have if we're going to move forward. I was heading the National Committee on Bio and Health Statistics at the time of the HIPAA started, and that was pre-internet and pre-genome. So obviously, it's not surprising that we have a lot of discontinuities at the policy level. I've enjoyed the presentations. Let me stop now and we'll pick it up.
Well, I think if we measure where we're at in the context of where we want to be, I'd say pathetic. Today we still face a lot of sort of very basic human issues, I think. It's organizational issues. I still find that in many institutions I meet, I have to introduce the genomic researchers to the phenomic researchers. So there isn't a connection. And certainly there's systems that support that research and the data that they have available to them largely not connected. We're not connected in terms of making that information then available at the point of care, as we talked about this morning. So we've still got a huge chasm to cross. Very much at the early stages. I think the awareness of the need for aggregation and integration of the information is absolutely core. So, in fact, I don't think the process of change will really take place until we recognize where we need to be and we all become committed to it. So, I think about it a little bit like the financial industry and the credit service bureaus. The early days of financial transactions were single events, and we're still thinking about sharing for research purposes or sharing for phenomic purposes where we're trying to get information to patients or clinicians as a single transaction, rather than thinking about it as a credit bureau where we need to bring together lots of information for the purpose of the deep kind of insight generation that we're talking about here and want to achieve. So I think, again, that awareness is the first step of the process, but we've got a lot to do yet to put the rest of the systemic transformation in place.
On the data sharing piece, one of the most important aspects is genomics today has one of the largest open source, not open source, but open data sets in the world. Right? Because almost 60% of the genomic research or 70% is funded by the governments in different countries. So that's one of the big assets we have in this space is almost 80% of the data is open already. With 1,000 genome data. You have the NIH, we know the sponsorships in the UK and Europe. You have ENPL. Minister of California, Santa Cruz actually has a genomic browser, which is again for free, that you can go type in stuff and find out what the variants are. So that's one piece. But the other piece is collaboration, because now that you have all this data at your disposal, a lot of the scientists potentially can collaborate with each other. But again, you've got to be careful in terms of security of that information or who has access rights to this information. And Intel, for example, may not be able to download these data sets behind the firewalls from a security reasons perspective. The second piece, of course, is one of the techniques a lot of the researchers are adopting are building these sandboxes called mine, yours, and ours sort of sandbox, in which I have my own data sets to play with, you have your own data sets to play with, and then we have a common data where we play together, again, based on this. So NCI just did a grant for the Genomics Cloud. It's a company called BioNimbus, I think it's a nonprofit out of Chicago. So there are a lot of institutions that are pushing for this open data.
Thank you very much, and I'm quite honored to speak on this panel here today. The interesting thing is when you think about data sharing and where we are with that, we are doing a lot of things as you just described and bringing the data around, but everything's being done in silos. And until we're going to be forced to ever be successful in data sharing or interoperability or anything along this way, we've got to make it where data flows like water. Now, if you go back and think about HITECH a number of years ago with the electronic medical records, we've made progress. We have basically taken paper processes, for the most part, and now made them electronic processes. But we should never forget that at this point, electronic health records are bit buckets. They capture information. We've not taken those bit buckets and started bringing together other great data sources and started bringing them into an analytic construct where they actually change and affect care at the point of care, because the most important time is when a patient and a doctor are sitting together. And if we have to have the right information with the right predictive ability, and we start looking at genomics and people's DNA sequencing, if somebody does have breast cancer, let's look at her particularly and find the right drug. So right now data is in a silo, and to answer the question, I think that we've got to do a lot more on the interoperability side if we're ever going to be successful, and that means bringing things together where people can actually use them and then make a difference.
I think the greatest challenge in sharing data lies in the fact that there are many bad data sets out there and we don't know how to identify them. So along with that, there's no really agreed upon standard for normalizing the data that we use. So NIH has put in a lot of money in generating patient data, sequencing data, and so forth, but the truth is that when I go back to my institution and try to analyze data sets, I encountered the problem of having to do everything from scratch again, meaning starting from processing of the data, filtering out bad data sets, and normalizing data in a format that I can actually use to infer meaningful findings about a system or some disease. So right now, I think sequencing is cheap. It's not as low as $1,000, but analysis of data that you get by paying $1,000 will be about $5,000 because I need to pay my postdocs and my students will spend about a month analyzing the data. So even though sequencing cost is dropping, the analysis cost is actually increasing. I think that's one of the biggest challenges.
Great. So I want to dig into some of these challenges that you've mentioned. So one that was brought up was this issue of data silos. This is obviously an organizational issue. It's a policy issue. How do we break through those silos? I mean, if we're talking about the goals in the earlier presentation, we say we can't wait till 2050. This needs to be 2020, right? How do we, in the next 5 years, really break down those silos so that the competitive advantage that organizations have is not around the data they have, but what they do with it?
I'll take a shot at that. I think, actually, I think the Clinical Translational Science Award initiative really has helped change some of that culture. But the culture, excuse me, the culture historically has been when the researcher gets their data and they tend to want to keep it. That's been the culture inside the organization. So you could get another R01 and stay around. So the norm is having to shift to you're a good person if you share, which is a new behavior that obviously is frightening to people who really spent their whole career kind of filling up those things. And so they're still in place. So I think, but the CTSAs have done a couple of things. I think one of the things they also did is, your colleague at the end of the table there has helped create essentially a set of people who are looking at how do you help develop research methods for dealing with some of this too. Because if the researcher is going to turn their data over to somebody, they want to know something's going to come of it. That's for sure. And it's got to be credible. And so I think the other thing that we've found is that we have a major research and development issue, as I think our last speaker said, and the data analytics side really on sort of the methods and so forth to be able to manage through that. I think we are evolving in that sense. What I'm not really quite seeing, I guess, that I'd like to see is how do we bring current public policy up to the concept so that the default setting is that we will share data unless somebody doesn't want it shared, rather than the default setting be that we won't share data unless every single person gets a knock on their door asking them to do it. And I think the fact of the matter is, I think most of the American public would rather have cures. And I think that's a challenge that I think that it being where we are right now with some of the kinds of senators and House representatives we have, I think we have a shot at actually really moving that needle. So I hope that we don't just see this as the technical fix that we need and the cultural fix in our institutions, but the citizen fix, which is going to take for us to also hitch it up and get up and do the work it takes to convince people that we can deal with these data securely and sensitively, and the same people you trusted to take care of you can trust to do research responsibly too.
Other thoughts on this?
I'll just maybe add to that. I think one of the things we can do is reframe the way we're looking at the problem, because I think today we're looking at the problem still in the sort of the lens of a data aggregation problem, privacy protection, as opposed to its application. So when we reframe the problem and focus on the outcome, which is what we're trying to do is improve outcomes with this integration of data. We're not trying to produce more research data sets or more research results to be published in PubMed. We're trying to find a way to get them into practice. And I think most people would recognize that increasingly is going to require more data. So when we think about the desired outcome, which is creating better outcomes, that the integration of data will become just a natural part of that. And I think we've been focused on the wrong place, which is hoarding in a sense. It's aggregation of data for its, for my purposes, not humanity's purposes. And I think there is something absolutely bigger here. And so, when we get that framing right, I think we'll enable it. The second piece is we've enabled that, sort of structurally within our healthcare system. So we talk about healthcare data belonging to a certain provider network. I can tell you from building analytics and big data companies and other industries that, and my last industry was the communication industry, that the data that AT&T has about us or the data Verizon has about us has no value to Coca-Cola. Okay? Coca-Cola wants and needs to know something about its customers across really the universe, all people. And so the idea that somehow AT&T owns something unique in its data or Verizon just isn't true. It's the same in the provider space today. Any particular healthcare provider, the data's not theirs. First of all, it's ours as patients, and second, it needs to be integrated in such a way to create better outcomes at the end of the day. So for that reframing and focusing on that integration, I think it's cool.
And just building up on that, I think you can work with Oregon Health and Science University and think about it this way, that you're an oncologist, you have 25 breast cancer patients, Sloan has 25, MD Anderson has 25. Could you create a system where when you log in to look for your the symptom, you get a list of 75 or 100, right? Completely identified, there's no source where the data is coming from, et cetera, et cetera. All it has is some fundamental things, right? I mean, potential markers, clinical trials that have been done, some history of some phenotypical history, and that's about it. Now, when you find your match, then you go get into a business model if you have to, right? Go to monetize information, or you get into a situation where you can, potentially add more content to that. But that will depend on the institution. But having built it once now, we believe that you can create such infrastructures. That's possible to do. But again, the big concern is having people actually sign up to play nice. That's always the concern, because people are getting into this evil notion that once you have this patient data, that is the new oil, as people are calling what big data is. That's an asset, and people are, if there's an asset, then you're going to find a way to monetize those assets, and that's a huge concern. Because what's happening with all the researchers and grants, that I think all the NIH grants, when they come out, the data has to be shared. I mean that's by default.
Animal data.
See, that's a clarification, so I haven't found that well now. So yes, so then could we make sure that every grant that comes out for a publication, because also we know that back at the data set problem, the quality of the information, right? I think about 50 or 60% of the Nature papers aren't reproducible. So even you get access to all those data bits, and back to your question, can I trust that information? Right? So we can create all the systems you want in the world, but standardized, if it's not normalized, not secure, I don't know what's going to happen.
And I think, going to your point about the need to be able to find the 25 patients here, 25 patients there, whatever it may be, there does need to be a way of actually doing research and cohort exploration that has a lot of multiple players. Because the problem is we aren't filling all the clinical trials. There's a lot of things that will never get to market quick enough because we can't locate them. We've created a cloud solution where right now we have a number of health facilities globally that do have that ability of putting some anonymized data in so they can actually go research and find out where are those patients that meet the following criteria and categories. It needs to be much more widely adopted and shared. But going back to the health IT side of the things and the interoperability and the issues in electronic health records, HITECH paid and helped fund for interoperability. And the problem is that Epic systems may not talk to Epic systems, Cerner systems may not talk to Cerner systems, and having them all talk to it to start communicating and interchangeably get the records, it's not going to happen. By having these multiple data sets, these multiple data constructs with the electronic health records, you are creating data silos. And so one thing that we need to do, and one thing that I hope the government will take up, is the fact that we need to come up with global-based standards and common languages and ways that we're looking at the information that's on the electronic health records. They should never be competing on the fact that they can create their own little universe and you go with XYZ and XYZ there, and that's the only way to communicate. That doesn't work. We need to have the ability where vendors compete on applications that are productive and actually help take the science forward, not on the data level that creates various entries and costs sometimes billions of dollars to implement.
I just want to add that NIH is putting in about $500 million for the next 4 years to fund 11 BD2K centers. BD2K stands for Big Data to Knowledge, and we are leading one of the centers at UIUC. And the first meeting was in November this year, and there were 11 centers in one place discussing how we can improve data sharing. And many things came out of that meeting. One of the things is to establish genomic API, as an application programming interface. So we will define features which every institution needs to identify when uploading data sets. And also, when we build software, we need to have API describing what our software does so that it can be ported to other centers.
So there have been a number of initiatives, very ambitious initiatives to collect and share genomic data at the Global Alliance Genome Project. When we're looking at the availability of data today, do we need to be focusing on that problem? Do we need to be focusing on how we create more data sets? Do we need to be getting that other 40% that's not open, more shared? Where should we focus our attention on?
What we call and how we define big data is not yet clear. But I would end up saying, even with as big as the data as we're talking about, it isn't going to be enough. I mean, what we've found out in the last 100 and some years, the human population has exploded. It's not only been productive, it's been very reproductive. And the human genome has become a much different genome than it was even 150 years ago. And not only that, we also find as you age, your own genome changes. And we also seen that the tumors' genomes change, or whatever thing you're talking about as well. So from my perspective, unless we're talking about really gigantic haystacks with really, really tiny needles, I don't think we're thinking on the scale at the policy level that it's going to take when I talk about making sharing the default. I think we're going to, at the minimum, have to have that. If we're really going to, because sometimes you get into these cell sets, they're pretty small, and that's where you also, big data can be big error. If the data curation and such. I mean, just looking at claims data for quality measurement, NESQIP showed that it's tossing a coin in the air. I mean, we're not even collecting the kind of data at the kind of granularity you need to make it SNOMED compliant or whatever to really find signal, not noise. So if that responds somewhat to your question, I think we really have to think about setting a pretty high goal and still have to deal with all the chaos that also may generate. But if we don't get the policy piece right for global sharing and such, I don't think we're going to get out of the gate.
Just a quick point, I'm looking at Peter there from the Global Alliance. The Global Alliance is working on an API. Right? NIH BD2K is working on an API. Are they the same APIs? Yeah. Perfect. So, so I guess these are one of the this is the key, right? Because the university and academic systems believe in the NIH process, but on the private companies, pharma companies, we go down the global alliance path. This is perfect that the two main things are talking, we have the same APIs, and that is a huge start for us today.
Regarding sample size, I have my own opinion about what's needed. So today we heard about increasing the number of sample size n for a fixed static capture of your genome, which is from the germline. In my opinion, that's not sufficient to actually capture the principles of biology. To do that, you need more comprehensive profiling of each individual cells in multiple regions of your body. Even in a tumor cell, we want to be able to profile comprehensive genomics, such as RNA, protein, non-coding RNAs, and so forth. So just genotyping a person is not sufficient to learn what is necessary to figure out the principle of biology. And that's what we are going for. By just genotyping individuals, you may be able to capture the variation. But in the end, human genome evolves. The variation will change. And what we are looking for is to learn how biology functions. And for doing that, I think we need to increase the level of complexity, not just the sample size.
So, earlier this year, NIH put out their new policy on genomic data sharing. And this seemed to be an incredibly positive step forward, especially compared to the way we traditionally approach data in the United States. I'd like to get the panel's reaction to both the policy itself, but also how do we make sure it's implemented well? Because as Don, you talked about, the incentives aren't really there for researchers to do this. So even though this is the policies come down to implementation. How do we make sure that the researchers are actually sharing the data in a useful way so that others get access to it?
Well, that's what gets among, there you start getting in the weeds of the IRBs. I mean, geographically, IRBs are all over the map, but unfortunately, their behavior's all over the map too. And what we've not seen is NIH has looked at, I think quite responsibly, to try to look at what kind of reforms can we make about IRBs and just data sharing and aggregation generally. But they've done it within the context of what current law is. After all, they're an actually legal piece of government. So they aren't the ones that are out there trying to figure out, well, let's rethink this whole thing and come at it really from the perspective of how we really want to get there. And at least I've been critical in the context that I don't think they're sort of half vision. It's good, it's better than where we've been, but I don't think it's moving us to where we really need to be. And I think even the research community out there would not necessarily disagree with that. As I say, they're living inside the government, so it's a little tough. Let me stop at that point.
Well, just before you stop, I want you to be really clear. So what do you think they should be doing?
Well, I think they are working with the 21st Century Cures Initiative. I'm not saying they're irresponsible. I don't mean that in a sense. I mean, they clearly have great understanding of what the issues really are. And I think they've tried to look at it in a stretch goal to some extent, but they don't push. Well, we've also had the problem with lack of sufficient leadership in the Congress and in the executive branch. I mean, at one point, the All-ONC office was taking recommendations on privacy policy without even talking about what the research implications, and admitting that they didn't even look at the research implications. So the fact is, if we're really not getting that kind of attention in our best of our system, how can we expect NIH to sort of make it right? I mean, I think it's not going to happen.
Well, I have an idea. So for those of us who rely on funding to survive, we need to publish. So instead of saying that you need to share data if you get this money, I think NIH should work with Nature Science and other journals and have the journals enforce the rule that if you're going to publish, you need to share data in a usable format. I think that would be much more effective in enforcing the rule.
Yeah, I'll just bounce back 1 second, because I do think actually some of we have yet to see the 21st century package of legislative bills hit the street. But I'm hopeful that they will talk about how do we start looking at some of this, actually for the first time sometime, hopeful. What I guess I'm concerned about is whether there'll be enough people get behind it, if they do say what we need to do to make sure that then the Congress acts on it and helps that. So everybody, all the stakeholders have a role, clearly, I agree. But the people that write the laws really are in a unique situation.
So I want to move on to the question of international data. So I think there's a number of issues here. One is, to what extent is the United States working well with international partners to collect and bring this data sets together, especially in Europe, because we've seen new proposed privacy regulations that would potentially have a really negative effect on the ability to collect and share data. And people would complain about New York's laws, but this is, it goes much more beyond that. To what extent are we able to integrate these data sets in different countries?
And I can give an example of EMBL, which is I think it's based in the Crick Institute in the UK. A lot of the NIH folks actually work very closely with EMBL and EBI, and they usually have the same data formats as well. A lot of the Japanese universities also follow the same guidelines as well. But I think just a quick point about the international data in the European Union, for example, patient data. There have been some laws now that if you are an immigrant worker, there are some provisions that you can carry your data with you when you go to another country. But you as a patient or consumer actually controls the data, and then you go give it to the thing. They are building analysis also here from Intel. European cross-border data sharing, those sort of laws are coming into play in the EU because, as you know, EU is growing rapidly, and because of all the new policies, immigrant workers are moving all over the place, and so naturally the data has to go with them. Those sort of laws are coming into effect. So maybe not just the data, but even some of the policies and laws could be shared across different continents.
You mentioned Europe specifically. I think the UK is really not the issue. In fact, the UK's privacy posture is much more sensible than ours. For example, they maintain that their nation's privacy people need to look at how other conflicting social goods meet also to the privacy issue. Our privacy also just has to worry about privacy. So the fact is, I think they, and also as you've seen, they see this as a huge economic issue for their nation. So I think the UK, in fact, is a chance to be a really solid player. I worry very much so about what the Swedes have done and what you're seeing in other parts of Europe, and also trying to essentially make data almost an individual by individual kind of approach with no social community good. And we are in it together. So I'm hoping that Americans will take that hopeful altruistic sense that I think is part of our spirit and act like Americans and actually lead this kind of sharing responsibly for the world. But there's going to be trade-offs, and rightly so. There's very good agreements at HHS on the data side with the UK, and to some extent with Europe as well. As we've heard, as you get to other nations, of course, some of those countries are easier to work with than others.
I'd say first we need to do with our European partners, because generally what we found, I think, is there's a real willingness to explore those privacy frameworks. What we find in Europe is privacy generally is a human right. Here in the United States, it may be a property right, a contractual right. It's the wrong one. We've got very different frameworks, and I think very much leveraging some of the learning from Europe about how to share assets is the first step. The second is, to your point about the UK treats their data as a national asset. And we're not yet thinking about it as a national asset. And when we do, I think our behavior and framework will change. So we tend to be constrained by the regulatory frameworks, all the things we talked about in terms of attorneys this morning and the limitations of HIPAA. We're not thinking about it as a national asset yet. And when we do, I think our behavior will change. So we spent $100 billion on the Eisenhower highway system to connect the country. We spent $100 billion getting to the moon. We spent $100 billion on EMRs now. But we haven't seen the asset value return, which is really integration and connectivity. That's what came out of the Eisenhower system. So when we understand it's a national asset, and we're going to need to share it globally. A lot of us are Europeans, a lot of us are Asians, a lot of us are Africans. So that broader data set's going to be important. So I think that there's absolutely the basis for in a sense, a global conversation on that, and leveraging some of the learning that I think Europe leads in.
Yeah, and as Ebola is showing us, there's no place to hide either. I mean, we are all in it. I mean, we are in it.
Yeah, we're also going to have to find something to do with the data sharing, just for the fact we're going to outputs-based reimbursement for our service. And so how is a pharmaceutical company actually going to go across borders and figure out if your drug is effective or not? And so from a research perspective, we're going to have to find a way to do it. And I'm right now tracking a very large innovation center for healthcare that's being opened up in Texas. Texas Medical Center is doing it. Simultaneously, Israel is about to fund a national research project and innovation center around translational medicine, security, and a few other very key things. Well, when you look at the fact you said we're a lot of different types of people, these are all have to be linkages made, so we can start seeing how we can drive the data. And there's no reason the Texas Medical Center and the Israeli Innovation Center and XYZ aren't working together, at least on anonymized data together. But we also have to worry about the question of clouds and cross-border data flows, which will actually facilitate it. And it's not our area of expertise.
There are a couple really big issues on the standard side. Standard came up, for example. ICD started as a top-down, as a way to try to make sense about etiologies and causes. But basically, we're much more SNOMED compliant. As people, it's from biology out. And when you're talking about really shifted standards and so forth, I think we're really talking about some really big China changes to reformulate this so that it's actually dealing with human biology. And it doesn't mean you can't still, you do need to know the etiologies and all those things too. But how that big tectonic plate crunch shakes out is just an example of the standards challenge we got.
So, building off the comment about Eisenhower and the highway systems, Fran Berman, who chairs the Research Data Alliance, has raised concerns about this idea of a lack of data infrastructure. And so, obviously we've been investing a lot in health IT, but is that a concern? Are we really, putting policy behind building data infrastructure like the UK, which sees this as a national asset? Are we doing that? Or if not, what would that look like for anyone?
I mean, the case in point, David brought it up this morning, is every country has a national 100,000 genome program. The Qataris are doing that. In the US, you have 15 of those 100,000 programs, right? Kaiser's doing one in California. Geisinger's doing it up in Shangri-La, as they call it, Danville. And then, I'm sure that there's something in Mount Sinai. Washington is doing something as well. So that right off the bat itself is off. Right? So.
I'm not sure that the whole architecture has been kind of looked at in terms of its moving parts. And obviously the focus today has been on genomics here, but part of this data ecosystem I'm talking about needs to be set up so that the patient, through a secure website interface to the record, the clinician, as they take care of patients, can look at the populations they're taking care of, but the data also go as appropriate for their professional competence, recertification and such, so they don't have to have the overhead of trying to manage that while they're also trying to see patients. I mean, we really need to look at who needs which data for what kind of social purpose and make sure it all integrates and also has a way of evolving over time, because it's not, like I said, these are complex systems. And all of this is going to have to shift and sort as it kind of goes. But if we don't have that thought through broadly enough, we'll never get the connectivity. Because our system, I mean, let's face it, if the government had said, let's go with Vista, which the VA system had a long time ago. When I was over in the UK at Cambridge, I was asked to look at their system. And at that time, I thought, gee, if you use Vista 2, we'd be a long way toward a national global standard. And of course, free economies like competition and all that, so guess what? We have 1,000 varieties. That's the way we do things in our kind of environments, and EPIC has become sort of epic in the ability to reach 100 million people, which is pretty impressive for a private company. But the point I guess I'm getting at is that we haven't had the will to also make standards and do it on the inexpensive side, but still very valuable. And let's face it, the VA was showing us the way on quality leadership at that point. It's had its trouble since. But I guess my problem is that the willingness to also lead when we can see it, but also see it more globally, is something that I think we undershoot where we could be.
So I think we're underinvested. I think we've spent the first $100 billion on a journey, all right? And so we're entering the era of computational health. That's fundamentally what's ahead of us. So I think we've taken the first step, which is the digitization. That's what the first $100 billion cost us. But in the era of computational health, we've got a lot more. And it's just not the portability of the data. So we don't have national infrastructure to do that. It's generally peer-to-peer today. So there really is no national emphasis. I've just been involved with Mayo Clinic in a NIH grant and some of the early review data that came back. Great idea, trying to make more things computationally oriented. Interestingly enough, the negative NIH feedback, just the reviewer feedback that came back, were all about practical limitations. Well, you can't do that in the cloud. Oh, security, it's an unsolved problem. HIPAA's not going to allow you to do that. None of those happen to actually be true, but I think our level of knowledge in terms of creating an infrastructure probably isn't there. I think we've, in a sense, undershot with our efforts on the phenotype side. So with the work of OMC, I think we're at risk of undershooting on the genomic side. So the good news is I think in the genomic world we're ahead somewhat. A lot more open data, a lot more open tooling. So the computational piece, meaning that the algorithms and the analytics have to be portable, the knowledge has to be portable, just not the data. And so in the genomic world, we've got a lot of the computational tools and infrastructure that are open source, that are being provided to accelerate learning. We don't see that on the genomic side yet. So the idea of a national roadmap, I think is a very good idea. And I hope the 21st century program begins to put that some of that thinking and infrastructure in place.
I apologize for jumping in again, but I wanted to, because next Monday, actually, I'll meet at the American Medical Informatics Association to help start building an interprofessional advanced clinical informatics certification program that you will have doctors, nurses, osteopathists, dentists, pharmacists, all of the health professionals being able, as advanced clinical informaticians, to help create this kind of integrated healthcare system over time. All these national organizations of educators believe that this is something we need to do. We've now just certified 500 physicians that have boards, and in the second year of that. So the point is, I think our workforce side of this is still not what it needs to be. And I think it's also true at the research informatics side, particularly on the genomic side, of how this fits to the genomic side. I think the human resource piece of this is also, these are great jobs. These are phenomenally great jobs. And I think we're underinvesting also on the human side.
Dan, you asked the question, do we have a lack of a national data infrastructure? And it strikes me there's two phenomenal global examples we should consider. First is Australia, the second is Singapore. Singapore has a national electronic health record under the theme, one record, one Singaporean, okay? Of course, their way of protecting data is more centralized, but no matter where you go in Singapore, your medical record, your information, whatever it is, whether it be health records or genomics as they're getting there, will follow you. Australia has a personally controlled electronic health record. It's a little bit different. Every state has their own repositories that get centrally brought together. It's a multi-layer system. Another good model for us to consider in the U.S. But in Australia, your health record follows you. Now, they've also made a change. Originally, it was an opt-in program. You had to say that you wanted to have data sharing. They now are changing it to an opt-out, so every citizen will be in it. Unless you say you don't want to be in it, then you can control how you do it. Now, what this has allowed Australia to do is they're already looking at very sophisticated decision support around genomics and putting personalized medicine and genotypes to effect to really cure diseases because they can bring things together. But they were very foresightful. They took them multiple years to get together to think about the legal, the regulatory, making sure they had the same snowman identifiers in the Australian version. But they were thoughtful. So do we have a lack of a data infrastructure in this country? Absolutely. Are we spending a lot of money? Absolutely. We should be a lot further along we are. But we should also look at Australia, Singapore, and other great examples in the world.
To underscore that, we don't even allow Americans to have a unique health identifier for research. Curation that improve both privacy as well as quality, security, and safety. Now, why we don't have an opt-out on that? For anybody that doesn't want to have one, fine, but let Americans have it. I mean, my gosh, most people want it. I mean, when I was on the President Bush's Interoperability Commission, the day we had the report out, a woman in the audience said, "My name is Mary Smith. You probably can guess where I'm headed." Was my question. I've gotten other Mary Smith's data in my record, and I don't want them getting this and confusing the doctors and having both care, privacy, and safety issues. That was a bunch of years ago. Where's that gone? So even for research, we don't allow the upgrade, the easier improvement. Do algorithms work? Sure. But you get about a 7 to 10 point jump in accuracy if you have that kind of a number.
So how do we move past this issue? Because, this is Washington. We focus on, instead of building solutions, we look at all the problems. And that's part of the reason, you go to these discussions and we get hung up on, you talk to somebody and say, hey, I want to cure cancer. They say, well, what about privacy? And, that's not the conversation we're having. We're saying, how do we advance the state of knowledge? How do we advance cures? How do we change this conversation so that it is about outcomes? And we put, we put these cures ahead of these other issues.
I'll be honest with you, one of the biggest ones is evidence. If you go to any physician conferences, they don't use the word genomics, right? And they've surveyed a lot of these international events where you have all the physicians going in, 5% believe in genomics, and 2% believe in genomics. So unless that tidal wave happens for all physicians, and everyone in the room here, I'm sure believes in genomics, but there's 95, 98% that don't believe in genomics. So I think as more evidence gets created through all the New England Medicine journals and things like that, just getting a publication in Nature, I think, is not good enough now. You need to have that evidence-based connection, and that's where things like phenotype data to sort of cross-code it is really important. But another point also is people are raising concerns that genomics is next, but there are 4 or 5 things after that as well. Right? This imaging will start kicking in now. Your cellular 4D pathways are going to kick in. What happens then? So there'll be more questions coming up. So let's be ready for all the answers.
God bless the patient advocacy groups, because, and particularly mothers that have kids with long-range disability. I think those people are interested in hope, not fear. And I think that's kind of the balance that we're talking about, is the hope of cure and the promise of cure. And I've practiced surgery for 25 years. I was doing dog heart transplants at NIH as a clinical associate. It's almost an outpatient procedure to replace somebody's heart nowadays. Not really, but it's astonishing. So I've seen this stuff works. Let's not talking about it doesn't work. So we have a great story. I think it's more at the emotional level. I just think we have to say, why does this suffering have to happen? Particularly when we're not having enough money to buy our way out of these things. We've got to work smarter.
And I want to open it up to audience questions. I just have one more for the panel before we do that. And that's, again, kind of around this issue of privacy. I've seen, especially in the last year, a lot of policymakers get hung up on this question of de-identifying data and having concerns about that. As you said, a lot of people don't even believe in genomics. So how do we change the understanding around that?
Sorry, I keep jumping up here, but I really am sorry about it. But anyway, I haven't thought about that. But anyway, the point is go ahead, somebody else.
I think we heard one practical solution today that I'm a big believer in. So I have heart disease. Mine was discovered by a 20th century technology, which was a heart scan. All of 3 minutes, non-invasive, and turns out it's exactly the rest of same as the rest of my family. So the 21st century solution to that is my genomic data and the ability to donate it. And so the idea of privacy, I believe there's not only a large but a growing sector where that contribution needs to be my choice. And it needs, 23andMe provided 23andWe, so the ability for me to contribute it. I sign my data consent form religiously, because that data, that both genomic and phenomic data, will be hopefully my son's ability not to have open heart surgery at 44. So that's a big change, and I think just anchoring on that, I think will go naturally. I think that's a great place to start. I think there's beyond that, we need to then reframe the problem again. We need to come back to understanding it's a national asset and priority, and build a framework for privacy evolution that accepts the importance of the information that to all of us as humans. So until we do that, we're going to continue to carry this baggage. Time to leave it.
Now can I add something? Yeah, I think the point is that we shouldn't use non-anonymized data if we don't need it. Sure, obviously, we want security as much as we can. But I think the fact of the matter is, it looks like, and people know a lot more about this than I do, colleagues that got in translational bioinformatics, said, you can run, but you can't hide. The closer and closer you get to somebody's genomic identifications, who you are. And I think we have an issue here of public credibility. A lot of this has to ride off of trust. And if we can't swear to the public that we actually can do this and still actually not involve them as who they are, and there's a risk, very small, but a risk associated with that. Most people are going to take that bargain, but I think we've got to be very careful acting as though somehow we can have all these other things and everything else and nothing ever has a problem on that kind of thing when 1.5 billion people are putting their genome on Facebook or something. I mean, it's just a la-la land kind of thing that we're kind of dealing with, I think, on this, where we have the people who are most capable of moving the needle on human health and suffering for individuals and populations can't get to the stuff to do the work, and they have to stand at the bedside and feel terrible about it because their local RFD puts barriers from them even taking care of their own patients, looking at their own data set. It's just wacky.
Hi, this is Megan O'Boyle, and apparently you've been reading my mind, because my laundry list before the panel started has pretty much been addressed. And I am a parent of a child with disabilities, so God bless you all for trying to solve the problem. And I think that it's going to take some time, but I think that the group that you need to find to answer this problem, to get the legislation, to get the Congress to spend the money, is the patients. We are highly motivated, passionate, invested people. And you know, some of the things that I jotted down before you started was our group is a very small, ultra-rare disease that was fortunate enough to get a Corey contract. And so we're collecting electronic health data. Well, we're not, really, because we can't get it from our doctors and our hospitals for various reasons that everybody in this room knows. People would rather pay the penalty, people don't scan our stuff, whatever. So that has been an issue. I've seen the IRB situation where our fabulous researchers are having their hands tied by overly conservative IRBs because they are taking what OHRP writes and going to the nth degree, and that's got to stop. If patients knew what was going on and how years and money is lost, they would be fanatical about it. And then the opt-out, absolutely. Patients should be sharing data unless they opt out. And we use a GUID, a globally unique identifier in rare disease. I think the autism community at NIH started it. But why aren't all newborns given GUIDs? And if their parents don't want it, opt out.
Do it.
And in terms of research sharing, I think that your idea about having the journal groups holding the researchers to the fire makes sense. But when is somebody going to publish the negative research so we can stop funding bad things over and over again because nobody knows that it's already failed? And then, I think that was it. Oh, one more thing. I think if you start with, maybe not the general public, but if you start with these foundations where there are sick people and caregivers of sick people, you're going to find this kind of passion. And I will tell you from a group, our children, there's maybe 1,100 diagnosed in the world. I counted 37 parents that had tattoos of their children's diagnosis when I was at the family conference. And they do, in fact, go from their geneticist to Facebook and say, my child's deletion is XYZ through XYZ. What should I expect? And so, you know, these rare populations aren't as concerned with privacy. And I know why privacy is important to a lot of our patient populations, but I think that if the patients can be engaged through the foundations, then you all will get the support that you need. And I think when the research community, the IT community is the one making the argument, it sounds like you're just being cold. So I think you have to engage the patients.
But I don't think it's privacy versus not privacy. I think we also want privacy. The issue is health is the first wealth, as Emerson said. Health is the first wealth. The fact of the matter is that's an issue. It's not that they aren't both good. They are good, and we ought to continue to see them as good. But health, ultimately, if you don't have your health, you don't have the capacity to help others have their health and so forth. It's just it's a wrong calculus right now. But thank you for your comment.
To your point about being a parent with a child with rare diseases, I'm the father, the son, and the husband of type 1 diabetics. And when my daughter became a type 1 diabetic at 5 years old, we said, there'll be a cure by the time you go to college. She's in college now. And you know what? That's unfortunate. And I had tears in my eyes when I told her we couldn't do it. And we were still working on it. But the point I also want to make is you talk about the people with tattoos and putting stuff out on social network. You can't even use social as part of the care process. And so we need to think about, when we think about big data and analytics and things along that line, there's a wealth of other things that people have put out there free and clear with no caring about privacy that we really can't use as part of the care process. So it needs to be expanded. So that's my
question about patients like me, that website.
Sorry?
Patients like me.
Yeah, we actually have our own registry.
Okay.
So we have a very specific thing, but it's, I mean, I'm all for patients contributing their data wherever they can,
Yeah.
but it shouldn't be in different places where it's not tied up. I mean,
Should start here in town.
So to follow up on her point, I've asked the autism people who pioneered it, who should make the national GUID? And they have no idea, so maybe I should ask you guys. Who should make the national GUID?
Actually, I've been working with a group called Shared Care and Cure that actually has worked with some folks that have a way of creating a unique identifier that's more like a credit card number. It's not like you're giving it as an SSN and it can't be changed over time. A person, if you have a problem with it, somebody take it, you can change it and so forth. There are methodologies now, standards out there. There are approved standards to do this. And yes, there's a small cost, there is a little cost, but look at the price we're paying by not having it. And on the differential side, I don't think there's any question. So the technology actually, I think, is really not immature at this point. I think it's just a matter of will, frankly.
And I think we treat it as binary. You're going to do it or not do it. And I think some of the discussion here is allow that as an option. I would choose it, okay, as a patient. So full stop. I lived in the UK and in Europe for 16 years, had a national health identifier. Best thing that had ever happened to me in the health system. So I think we treat it as something very evil or something very good. I think there's at least a good option in there, and I think it needs to be the national agenda. It needs to be.
Sure, I mean, all of those are good points, but the problem is, if we have 600 places making GUIDs, then they are unlikely to be transferable. And the technical aspect is doable.
But there is a technical framework for that.
It's fairly easy to let distributed groups who agree with each other that they're going to do it to create GUIDs that are not going to overlap.
Joel, are you putting this up on the website later? Excuse me, I apologize. On the finish. Are you putting this up?
The video.
Because I can actually put that standard up. I'll get him the information, so at least everybody here who wants to look it up and know about that story.
This is exactly the dialogue we want to foster, guys. This is going right into the roadmap, and this kind of discussion is perfect. So yeah, we'll put it. I can get it.
yeah, everything will be posted.
I've got a question on economics. Who's opposing this? It's great to have this group of data wanted users, Oracle, IBM, Intel, all of you in research. But there is an economic opposition to this. One is the business model of selling data, which scares the hell out of me. With Cleveland Clinic, MedStar selling data through Explorers, it's unbelievable. We should be paying for doing analytics, not for selling data. But the incentives aren't there. The same with the academic institutions. At a SAS meeting, talking about the sharing of the pharmaceutical research now, which pharma and EFPIA have been pushing, and it's come about this January. The question I asked one of the researchers at Duke was, publish or perish. You don't want to share your data. He says it's worse than that. At academic institutions, you don't want to share your data because if you don't get the grant for using your data, somebody else will, and you won't get paid. And you've got a 1 in 20 chance today of getting a grant out of NIH. And sequester is going to kick in again, as I am told, next month. You know, and Europe is way ahead of us. They're spending money, but there is opposition here. You go to United, and you go to Optum, and tell them that they're going to give their data away for free from now on, and wait till you hear that response. Thank you.
Just maybe, if I may push back a little bit, which is, there's monetary value to data. Okay, it happens in every industry, and I don't think we ought to be shy about it. It takes an enormous amount of infrastructure to capture it, process it, normalize it, and there's value in that. And so there's some aspects of monetary value. The question is whether or not we can do that in an efficient way to allow that value to be released, because we've got to find, and I absolutely agree with your point, that it's the analytics on it that create value in the business process. So it's the thing that reduces preventable adverse events, it's the thing that improves outcomes.
But we're paying for it when we pay for our medical insurance. And we have paid for that creation of those databases. And they should belong to us. And when the Jason report, and here's Jason number 2, go read it, okay? They said the data belonged to the patient, and they got slammed. It really should be our data. We've paid for it, and we ought to get the benefits for it. But as Don points out, we don't look at the social value, and it really belongs to everybody. And I hate to sound like a Democrat because I've been a Republican all my life. Okay? But it's our data.
I want to go off on a little bit of another point, and he wants to get 2 more questions to us. But it relates to SNOMED. Actually, SNOMED really is a model that we could use. It was started privately by a group of pathologists as a nomenclature. When Tommy Thompson was secretary, he said, we will license this to anybody free across the country. Ultimately, they bought that out. The same people internationally now manage it. And I think the same thing. If we're close to where Epic becomes the best horse on the street, we ought to buy her out, pay Judy whatever it costs, fair price, she deserves her money, but then also put them and others in charge of maintaining it for us and make sure it's international as well, because they're doing work internationally. And let's start doing things that make it interoperable. And we have social mechanisms of being able to, I think, treat business fairly. There's clearly cost and a lot of energy and sweat, blood, and tears went to get those social goods. But if they become now a utility need for the whole world, we ought to be able to figure out a way creatively to do that.
This is where the EMR companies need to come up with a common standard, common way of storing their data, so it is not an issue of data. It's something we can transfer. And then you look at it on the application layer, they can all compete on the application. There's no reason the switching cost should be $1 billion.
I want to get we have 2 people who have been waiting patiently for a question, so why don't we get both of them and I'll let the panel respond and then we'll have Dennis.
Okay, my name is Peter Goodhand. I'm the Executive Director of the Global Alliance for Genomics and Health. So thank you, thank you to Keaton for the call out to us. But we spent the last 2 years on this left-hand side of the curve of trying to globalize the generation of knowledge. So first of all, kudos to the panel, because everything they said about interoperability, I think is right on the money in terms of the way we can do the knowledge generation. I think where we have a lot of challenge, and we've heard it more and more today, we heard it on Monday, is that the clinical application, clinical implementation is healthcare system based. And so whether it's a national system, a regional system, an insured system, an integrated system. And I think when we get to that, the way this genomic knowledge will be translated into health at the community level is very much reflective of the current healthcare systems. And that's where the globalization piece changes. I think the health, we talked about health records and how reflective they are of, say, Singapore. We often use Denmark as an example. But Denmark's not that different from Kaiser, except Kaiser's twice as big. So I think one of the challenges you have in the US is that the country is so big and there are so many options. It's harder to mobilize 300 million people than it is 5 million people in Denmark. And the other comment I would have is just echoing that last comment from rare disease groups. What we're seeing is the voice of the patient, particularly the rare disease families, who are globalizing this agenda. They're not prepared to wait for all those rules and all those barriers. They are moving it. So what we've done from the beginning is make sure we had that voice at the table, because that's where the sense of urgency comes from. And I think beyond the rare disease groups, we're going to see more people like Paul. So I'm sorry that was all commentary rather than questions, but what I heard is exactly what I've been hearing for the last 2 years. It is moving in the right direction. And finally, America is showing leadership. The Global Alliance wouldn't happen without American leadership and initiation. And people and organizations like NIH are doing the right thing through BD2K, and they're trying to do it on a global basis, and that even extends to Africa. there would be no genomic research in Africa if it wasn't for NIH and the Wellcome Trust. So thank you. You're doing great stuff.
And my comments kind of build on that. This is Claudia Williams. I guess one thing that's palpable is the sense of urgency and frustration in the room. I just want to offer a straw man, though, for a way forward. I'm not sure we're going to be able to take the kinds of steps that Sweden, or maybe that's not an example, Australia, Singapore, UK did. We have a very different healthcare system. We have a very different set of norms. We have a very different role for government. And I wonder, though, given the size and complexity of some of these large delivery nodes, if the kind of approach that PCORI is taking, where you have large delivery systems with huge research capacity, you're able, with a sense of trust in that community, to collect that data. You tackle the problems around IRB, you make consent a lot easier, you engage patients at the front end, and then you figure out a way to share across those nodes, versus, I'm just not sure a big bang approach where we imagine some global thing is going to work for our nation. So that's one. I think the second thing I've heard, and we actually had a great meeting this summer with a lot of the patient data groups to talk about data donation. And one thing that came out really strongly is that patients are also fed up with the framework of privacy as the negotiating point. They want to negotiate over a lot more. They want a social compact around what happens with the results. They want to access both their own data and the results of that data. And frankly, sometimes other folks are the ones saying what you care about is privacy. They often care about openness, about transparency, about liquidity of the data, and ability to actually inform what research focuses on. So I just would offer that I agree that privacy is sort of a limiting framework, but patients want a different framework, but are often told, no, you're the patient, we'll take care of that. So I think we have to think hard about how to have a different relationship with patients who would like to donate their data and participate.
I respect that a lot, but I like to push back the stance. I don't see why we can't try to do both. University of Virginia has been getting a lot of press recently, not the kind that you usually like to talk about, of obviously Thomas Jefferson and James Madison and James Monroe. But my avocation is American history. I don't think it's a stretch too far. I don't think that's what the people that started this country, and George Washington and John Adams and all these people had in mind. I mean, they gave us a system. We can make it. We can make it. And I don't think it's either or. I think we ought to do that. But what I'm saying is we're talking about the human genome is a very complex thing. And if you can't scale, some things you may make progress on. But I think we should do both. I mean, I think it'll take both. And I guess all I would ask is let's do both. I mean, let's work and do both.
Are we doing one more question? Or quick? Yeah, quick.
Very quick question. So if anybody is looking for a great Christmas gift, there's a book called The Big Roads by a guy named Earl Swift that is the history of the highway system. Doesn't sound very interesting. But really there were three foundational aspects to it. One was the fundamental question is, are we going to build a national highway system, a top-down from point A to point B, New York to San Francisco, or are we going to build a bottom-up system of national highways? The second was, what is the governance model for this? And this actually was something established way back in 1919, along with that decision of having, again, a bottom-up federal-private partnership where state highway organizations took, the third piece, the financing model, where we had a gas tax, which is starting to get less than worked well for a long time, where the states actually would divvy up the number of miles of road and the dollars that the federal government came, with the caveat that they would follow standards, and worked all of this out. It took a long time to put together because without that it wouldn't work. And in the 50s, when we finally did get funding for it after World War II, the plan was on the shelf and you could just execute. So The Big Roads, Earl Swift, great Christmas gift.
Thanks for that. I think that's a good place for us to end here. So
about 8 months ago, this whole day, and we really didn't know what it would shape up to be, but we are just so delighted with the input, the energy, the speakers, the panels, it's just been really everything we had hoped it would be, a really open discussion. Thanks to Joel and his very talented staff, and to Daniel and their staffs for doing just an amazing job on today's event. And just, final few words. We've talked a lot about data. Size, velocity, it's only important if it moves. Cost, we talked about the genome going from $1 billion to $1,000, even faster than Moore's Law. The value, ownership, protection, and all of these issues are ones that we really need to deal with if we're going to make that goal of having genomic information in clinical practice by 2020 rather than 2050. So we ask you all to work with us as we develop the white paper, the recommendations to Congress, and continue to have this interactive dialogue as we move this forward. So thanks so much.
A few words. Between you and a beer. Just a few quick words, process words. Thank you to our sponsors. Thank you to Daniel Castro and his team. Thank you to all our speakers. So at the beginning of the day, I said if you're looking for a dog and pony show, you should leave. I hope we've gained some important insights. I know I have. My sense is we are going to want to do this white paper that I talked about at the beginning of the day. And I think it's going to be kind of the same type of format. A third talking about the problems, and 2/3 talking about the solutions. I'd like to make it characterized by three things. The first, I want it to be crowd-authored. So if you have ideas, if you have thoughts, send them to me. It's jwhite@healthitnow.org. Or so anyone on our team, you just go to our website and find us. But we want that input. I think we want to make it very collaborative. And so we're going to be reaching out to FDA, some of the folks that weren't here, but that we did invite. So FDA, the HHS secretaries people, other folks who are not here, to make sure we're getting their input as well. And then the third thing is we're going to do it quick. If you didn't get the sense from this last panel, these guys are going to be moving quick. 21st Century Cures with SGR, with some of the other vehicles that's moving. I don't want to miss an opportunity. What I learned today was there's lots of opportunities that we can capitalize on. So let's do that. Let's get this thing done, and then let's start advocating for the things that we believe in. Thank you very much for being here. I appreciate your participation.